A very good morning to one and all present here. On behalf of Seva Sadan's R.K.Talreja College of Arts, Science and Commerce, Ulhasnagar, and the Microbiology Society India, I, Kuldeep Mhatre, extend a warm welcome to you on this second day of our two-day international conference on AI and in silico insights in biology. Day one was truly enriching with inspiring addresses, enlightening keynote lectures, and vibrant technical sessions. Today, as we step into the concluding day of our international conference, let us carry forward the same enthusiasm, curiosity, and the spirit of learning. Albert Einstein once said, the measure of intelligence is the ability to change. In many ways, artificial intelligence and in silico approaches embody this idea. They are reshaping research, opening new possibilities, and guiding us into the future of biology. Before we begin the academic proceedings of second day, let us dig into the blessings of Lord Ganesha, the remover of obstacles and harbinger of wisdom, success, and new beginnings. I request everyone to join their hearts and minds as we begin with the Ganesh Mandana. I now request our technical support to kindly play the Ganesh Mandana.
गणनायकाय गणदैवताय गणाध्यक्षाय धीमहि गुणशरीराय गुणमण्डिताय गुणेशानाय धीमहि गुणातीताय गुणाधीशाय गुणप्रविष्टाय धीमहि एकदन्ताय वक्रतुण्डाय गौरीतनयाय धीमहि गजेशानाय भालचन्द्राय श्रीगणेशाय धीमहि.
Thank you all for the soulful invocation. With the blessings invoked now, it is time to begin our academic journey for the day. We are honored to host today's keynote lecture in silico sparks in pharmaceutical research to be delivered by Dr. Anil Folkmore, a distinguished industry speaker whose insights bridge the cutting-edge science with the real-life applications. May I now invite Dr. Neeta Gundavar, Madam, to kindly introduce our keynote speaker. Ma'am, kindly unmute yourself. We are not able to hear you.
Yeah. Sorry, sir. A very good morning to each one present here. It is my privilege to introduce Dr. Anil Folkmare, an eminent professional in the field of pharmaceutical sciences, whose distinguished career spans more than 28 years of expertise in quality control, quality assurance, and global compliance (CQA). Dr. Folkmare holds a doctorate in microbiology and has worked with several reputed multinational pharmaceutical companies. He is serving in leadership capacities such as CQA and global quality at organizations including Nicholas, Agenta, Cadila, Acombs, Alembic Strides, and Aurobindo, USA. His vast experience covers both sterile and non-sterile formulation facilities, and he has successfully handled global regulatory audits from agencies such as US FDA, UK MHRA, EU, WHO Geneva, MCC South Africa, PICS, and Zizobona. A passionate researcher and academic contributor, Dr. Folkmare has published 30 research papers in reputed international and national journals, authored 4 books, and has been an invited speaker at more than 20 national and international conferences, including those held in the USA, Russia, Bali, Maldives, and Uzbekistan. His academic involvement includes conducting training programs and mentoring in the field of pharmaceutical regulations, thereby creating a strong bridge between industry and academia. Currently, Dr. Folkmare is serving as the Director Technical at Omnicals Pharma Limited, India, and Omnicals Pharma Unipesol LDA, Portugal, where he continues to contribute to advancing pharmaceutical standards and practices globally. We are really deeply honored to welcome Dr. Anil Folkmare as our keynote addressee today. His visionary leadership, profound knowledge, and global perspective promise to inspire us all. I now warmly invite Dr. Folkmare to deliver his keynote address. Thank you. Over to you, Folkmare sir.
Thank you. Thank you so much, madam, for invited me as a speaker in this international conference. So, my today's topic is in silico sparks in pharmaceutical research. As you know that India is one of the leader in pharmaceutical sector, and we are the topmost country to provide the generic medicines across the globe. Can you see my? Yeah, madam already introduced me.
Yes, sir, your screen is visible.
Yeah. So whenever we supply any pharmaceutical medicines to any country, they require their approval. For example, in USA, if you want to supply, then US FDA is required. In United Kingdom, UK BHR is required. Europe, European approval is required. So like that. So these are the some photographs. And this is my book on regulatory audit observation and style of responses. So coming to bioinformatics. So it is a scientific subfield that involves, just a minute. So that involves using computer technology. As you know that because of computer technology, it helps us to store the data, to analyze the data and biological information in pharmaceutical products. So it will be really helpful for scientists or clinicians use databases that organize and index such biological information to increase our understanding of health, diseases, and certain cases as a part of medical care. And it is a fast emerging field in pharmaceutical industry. And this field, bioinformatics, was widely used earlier in other branches like biotechnology, biology, and many other fields. But nowadays, finding a wide application in pharmaceutical sector. The work described different aspects related to application of computers in drug designing, discovery, and drug development, like formulation designing. And bioinformatics is the combination of computers, databases, statistics, graphs, and 3D plots. Make the big problem of designing the new formulation of total new molecule is the most interesting aspect of the field in the pharmaceutical field. And any new drug development is a time-consuming activity in pharmaceutical industry. So that's why optimization process, which helps to reduce the number of experiments to be conducted and help the research scientist, specifically pharmacist. And also bioinformatics helps to link the information often from different fields. So we, in our pharmaceutical field, we are using different softwares like design is state is design experts. And these are available in the market for all the aspect in design the formulations, makes the costly and time consuming process very easy, fast and cost effective. So bioinformatics is the branch which deals with the application of computer technology for the management and analysis of the biological information. Bioinformatics is the rising sun in the global as well as for Indian pharma sector. This rising sun can produce immense economic hit for the growth of the Indian pharma sector. And during pandemic, you have seen that the rising sun of Indian pharmaceutical sector. It is being an interface between the modern biology and informatics, which is involved in discovery, drug development, and implementation of biological process with the goal to serve and help in healthcare sector. In the pharmaceutical sector, it can be used to reduce the time, cost, and make the people understand. So these are some important pharmaceutical areas and the utility of bioinformatics technology. So four means drug discovery, designing, development, formulation development or product designing, polymer modeling, and pharmacology. So if you see under drug discovery, only 10% of the drug molecules which are identified in research area make through the drug development phase. It means many of the potential drugs do not make it to market. An expensive time, resources which has been invested suffer a heavy loss without generating any capital. Simulation and informatics can significantly help in improving the efficiency of drug development, cutting cost and improving margin. If you want to develop one drug, so at least 3 to 5 years required when it will come to market. There are many bioinformatics resources which are growing importance to understand and predict absorption, distribution, metabolism, excretion, and toxicity of both existing drugs and potential drug leads. So formulation designing, when we do, so that time formulation is the process of mixing of ingredients. So when I say ingredients means active ingredient as well as non-active ingredient in such a way that produce a new or improved product. The formulation department must balance the different marketing and deliverability requirement with chemical constraint and cost to come up with the best possible drug delivery method at the best price. The information regarding the number of batches which are formulated in the designing process will be stored in the corporate database, which will help the scientists to access the result and methods from anywhere in the world. So minimum 3 batches required during the designing product formulation. So more than 3 means 5 batches, 10 batches sometime required. So that's why this information is helpful from the corporate database. So they can use their time and resource on new research rather than repeating the old work or traditional work. Also with the aid of optimization software, the best formulation can be obtained by using the data of the previously formulated batches. In polymer modeling, so many of the drug delivery devices are polymeric, with drug either solubilized or emulsified in the polymer. This drug delivery system have a mesoscale structure between 10 to 1000 nanometer. The amount of computing power required to model this system at an atomistic level is prohibitive and macroscale technique, such as finite element analysis or computational fluid dynamics. So this finite element analysis is a computer-based simulation method that predicts how complex product and structures will behave under various physical conditions such as stress, heat, or vibrational. And this mesoscale modeling, focusing on the nanometer length scale, is helping scientists or pharmacists to develop colloidal delivery system for drugs. Achieving a suitable formulation of the drug candidate can determine the rate at which a drug gains FDA approval. So I told you in earlier slide, when we supply any medicines to any country, their local FDA approval is required. And this FDA approval process also lengthy process, sometime 6 months, sometime 2 years also required. And techniques such as polymer modeling are helping to create a new methods of drug delivery. Pharmacology. Bioinformatics is used in drug target identification and validation, and in the development of biomarkers and toxicogenomic and pharmacogenomic tools to maximize the therapeutic benefits of drug. So when we do the drug discovery in pharmaceutical, it is a step-by-step process by which new candidate drugs are discovered. Traditionally, pharmaceutical companies follow well-established pharmacology and chemistry-based drug discovery approaches and face various difficulties in finding new drugs. That's why it is a time-consuming process. In the highly competitive pharmaceutical industry, the first company to patent a new chemistry entity for a specific treatment takes all the spoils, leaving other competitors to mostly wait for patent expiration. Almost patent expiration period is 10 years. Nowadays, therefore, pharmaceutical companies invest heavily in all those approaches that show potential to accelerate any phase of the drug development process. So one of the major thrusts of current bioinformatics approaches is the prediction and identification of biologically active candidate and mining and storage of related information. Drugs are usually only developed when the drug target for the drug discovery is increasing exponentially. Mining and warehouse of the human genome sequence using bioinformatics has helped to define and classify the nucleotide composition of those genes which are responsible for the coding of target protein, in addition to identifying new targets that offer more potential of new drug. So, drug target validation is one of the major and important activity in pharmaceutical sector. So, bioinformatics also provides strategic and algorithm to predict new drug targets and to store and manage available drug target information. So after the discovery of potential drug target, there is an interest. The establishment of such a key association provides justification for the drug development process. This process, known as target validation, is an area where bioinformatics is playing a significant role, and drug target validation helps to moderate the potential for failure in the clinical testing and approaches phase. So if you see the drug target validation, you can understand through this drawing. So gene sequence from HGP. So that is a human genome project. And this international scientific efforts aim to map a sequence all the DNA in humans and determine in order of all 3 billion base pairs and identify all human genes. So bioinformatics related genetic information, human functions. Then therapeutically relevant protein molecule target. Parallelly, this combinatorial library help us for high throughput screening, HTS, and then hit identified. If you see optimization lead, hit to lead activities, then focus on combichem libraries, hypothesis driven drug design, and serial synthesis. So before coming in new drug, so there are the scale up batches, patent, admit, formulation, safety, clinical trials, and FDA approval. So this is the sequential chronology activity for drug target validation. So if you see the clinical stage is very important activity for drug development. So there are 3 major phases during clinical trial: phase 1, phase 2, and phase 3. So phase 1 trial is to assess what dose of the drugs and drugs the human body can tolerate and may assess an element of drug effectiveness. Phase 2 trials test the drug in the relevant patient population. Exception are oncology drug and orphan disease therapeutics. So often there are 2 parts, phase 2A and phase 2B. Phase 2A trials test a smaller number of patients. And phase 2B is drugs are tested on a larger group of participants to further evaluate their efficacy and safety. So it is a common for multiple phase 2 trial to run simultaneously, testing various clinical indications, combinations, or formulation of the drug. And finally, phase 3 trials in this trial, researcher compare the trial drug to current drug already available to treat the same condition. The trial drug must be better than the current drug available to pass phase 3. For example, the new drug must be more effective or have less side effect with respect to existing drug. So bioinformatics is always beneficial to pharmaceutical industry during these clinical stages. So because all these 3 pages generate the data, often in vast amount, this is where bioinformatics come in. Bioinformatics analysis can be applied to data from primary outcomes as well as each of the following data which can be generated during clinical trial. So genotype or clinical phenotype, flow cytometry, hematological test, HLA typing. Then pharmacokinetic is one of the important measurements. And these are some benefits in pharmaceutical industries. Applying bioinformatics approaches to clinical trial data ensures we reveal all the key insights in the data. In turn, these insights can inform the next step and ensure maximum returns on investment in pharmaceutical research. So bioinformatics focuses on 2 main areas, data management, data analysis, and which find application in helping scientists or researchers in fast research, leading to quick inventions by providing readily available information with the help of computer technology. In interlinking information from different fields and leads to quick results, and in designing information available on paper or in the form of specimen. So these are some references. Yeah, thank you. And first slide, we have started from Ganesh Vandana. So Ganesh Vandana, if you see, one message is there. Lord Ganesh is having 2 big ear and small mouth. It indicates that listen more, speak less. Then Lord Ganesh is having small eyes. It indicates that we should do microscopic observation in and around us, whether during research or during our day-to-day activity. Lord Ganesha is having 4 hands. That indicates that we should do hard work. Without hard work, we cannot get any result. And Lord Ganesha is having patience, stomach. So big stomach indicates that we should have patience. If you see all these 5 factors, if you have, then we can do a significant contribution in our research area. So thank you so much. Yeah, over to organizer.
Thank you very much, Dr. Fokmare, for the enlightening lecture. Your talk has beautifully highlighted how in silico approaches are igniting sparks of innovation in the pharmaceutical research. Thank you so much, sir.
Yeah, thank you.
Now it is my privilege to invite Dr. Nidhi Mahendru. She is currently serving as an assistant professor and head of the Department of Biotechnology at Guru Nanak Khalsa College, Haryana. She is also a valued member of the advisory committee of this conference. She also holds the esteemed position of State President of Haryana Unit of Microbiology Society, Indiana. Dr. Mahendru, we are honored by your presence, and I request you to kindly share a few words with us. Dr. Mahendru?
Hello.
Yes, ma'am.
Yes, sir.
You're audible.
Am I audible?
Yes, ma'am.
Okay. Good morning, everyone. Respected dignitaries, esteemed speakers, faculty, and dear participants. As an advisory committee member, it's truly an honor to be a part of this international conference on AI and in silico insights in biology. The past 2 days have highlighted how artificial intelligence and computational tools are not just supporting biology, but transforming it. From drug discovery and genomics to environmental and healthcare applications, which makes this gathering really a special and is the spirit of collaboration of academia, industry, and young researchers. Together, we are shaping a future where innovation in biology is accelerated by the power of artificial intelligence. I congratulate the organizers for creating such a vibrant platform. Thanks to the speakers for sharing their invaluable insights and appreciate all the participants for their active engagement. Let us carry forward this enthusiasm and continue building bridges between biology and technology for the betterment of science and society. Thank you very much and have a very good day. Thank you. Thank you, Professor.
Thank you so much, Dr. Mahindru, for your encouraging words. Your guidance and support as a member of Advisory Committee have been invaluable in shaping the success of this conference. Thank you, Madam. We are also privileged to have amongst us Dr. Aruna Biswas, State President of West Bengal Unit of Microbiology Society of India. She is currently serving as Assistant Professor and Head, Department of Microbiology at Raidigi College, West Bengal. And she has also contributed as a researcher at prestigious Albert Einstein College of Medicine, New York, USA. Madam, I kindly request you to deliver a small talk.
Am I audible? Hello, am I audible?
Yes, ma'am. Yes, ma'am. You are audible.
Okay. Thank you, sir, for giving me this opportunity. Good morning, everybody. It's an honor to be a part of the advisory committee of this two-day international conference on AI and in silico insights in biology. I'm really thankful to the organizers, Seva Sadan's R.K.Talreja College of Art, Science and Commerce, Ulhasnagar, and to MBSI, especially to the National President of Microbiology Society India, Deshmukh sir, who I think actually recommended my name. And so these 2 days are really an enriching journey for all of us in biological sciences, because AI is such a cutting edge tool, and it's going to transform so many zones in our biological field, from agriculture to environmental sciences to, of course, our health sector, because, as we heard today, how it's bioinformatics and AI, they are crucial to drug designing and health management and medical management. So thank you, organizers, for this wonderful conference, and thank you for allowing me to be a part of it. Let's again continue our enriching journey. Thank you.
Thank you so much, Dr. Biswas. With this, we conclude the keynote session. The conference will now resume at 11 a.m. sharp with the technical session 3 of paper presentation. We request all chairs, judges, and participants to kindly reassemble on time to ensure the smooth and productive session. Dear participants, there is one more instruction. The feedback and consent form, the attendance form, it's shared in the WhatsApp group as well as in the chat box of this. It is a common form for both the attendance as well as feedback. So I request all the participants to kindly fill the feedback form.
Hello, Patil sir, can you hear me?
Yes, ma'am. Yes, ma'am.
Sir, can we take one picture? We can ask everyone to please switch on your
Yeah.
camera. We
Yes,
can
sir.
have one picture.
Okay, okay.
And even make Kuldeep sir as co-host so that even he can. Yeah, okay, he's there.
Okay. Kuldeep sir, kindly announce one time only.
Smile also required, Dr. Ranjana, during screenshot.
Try.
Dear participants, I request all of you to kindly switch on your camera. Okay, we have changed the settings so that we can have a group photo.
Is it done? Thank you.
Wait.
Yeah, thank you. Thank you. I think it's
Yes,
we
done.
have taken enough screenshots.
Done.
Okay, dear participant, if you want to take a break, you can take a break. We'll be resuming our technical session at 11 a.m. Thank you.
Good morning, all. We are ready to begin the technical session. I will first take this opportunity to introduce our esteemed judges for the day. We have with us Kuldeep sir. The judges have joined, I suppose.
Hello, hello, Physics sir. Dhananjay sir asking for the meeting ID and passport. Can you please forward? I could not. Uh, I'm not getting where it is located.
Nishad sir, did you say something? Please repeat.
Yes, ma'am. Mankar sir is asking for the meeting details, that is ID and passport.
Yeah, it is there with our link. I will just send it to you. It is there in the group too.
Yes.
Just a minute, I'll send it to you. Or I'll post in the group again, Nishad. I have sent it in the conference group, Nishad sir. Meeting ID and passcode just now.
Okay, thank you.
Patil sir, let us know when Mankar sir has joined.
Yes, ma'am.
Participants, I'll take another minute or two till Mankar sir joins.
Yes, ma'am. Now Mankar sir is with us.
Yeah. So
Good morning, all of you.
good morning. Good morning, sir.
Good morning, ma'am. Am I audible to all of you?
Yes, sir. I'll take this opportunity to welcome all of you back again after the session 1. And this is my honor to welcome our judges for today. I will take a minute to introduce the 2 judges, then we can start with the presentation. Our first judge for today is Dr. Dhananjay Mankar. He is an assistant professor at Center for Hospital Management, School of Health Systems Studies. He has done his diploma in Information Technology too. He is a Bachelor of Homeopathic Medicine and Surgery. He was an academic program manager at Center for Hospital Management, School of Health System Study, Tata Institute of Social Sciences, Mumbai, India. He was also a research associate with Center for Hospital Management, School of Health Systems of Studies, Tata Institute of Social Sciences, Mumbai, India. He has been a consulting homeopathic physician at Ma Bhagwati Homeopathic Clinic, Akola, Maharashtra. In his Google profile, his H index is 2, and he has published 22 articles in reputed international journals. He is a author and co-author for 2 books. He has published 9 chapters. He has in digital print press 4 articles of his. He has attended and presented in more than 16 research conferences at international level. Sir, we are very proud and honored to have you with us today. Welcome, sir.
Thank you, ma'am. Thank you. Thank you very much.
Our next esteemed judge is Mr. Nishad Sanjay Supugade. I am proud to say that he is alumnus of our alma mater, and it gives me immense pleasure to introduce Mr. Nishad Supugade. He is a motivated and research-driven postgraduate in microbiology with hands-on experience in academic research, in silico studies, molecular biology, and scientific communication. He is committed to fostering an engaging academic environment while promoting scientific inquiry and interdisciplinary thinking. He has 2 research publications to his credit with 6 presentations. Technically, he is skilled in microbial culture, staining, biochemical tests, antimicrobial susceptibility testing, in silico tools like Pyrex, Autodocs, Vina, Discovery Studio, GrowMax, and software like Microsoft Office, Google Workspace, programming tools like Basic R for data analysis and visualization. Mr. Nishad, it gives us great pleasure to welcome you too, sir. With this, I will request the judges if they have any particular instructions for the participants. We have already instructed them. The presentation will be of 5 minutes, maybe extending to 1 more minute. Questions from the judges, 2 to 3 minutes. So we have a total time of 10 minutes for them. We will be proceeding as per the list shared to you as well as participants. I will be interrupting at the end of 5 minutes to the participant so that they can wind up and come to their conclusion in 1 minute. The videos for the participants when they come to present will be on during the question and answer session also will be on. I request judges if they have any instructions to be further relayed.
No such as actually. I think they are already oriented well. We can proceed with that.
Thank you, sir. Mr. Nishad.
From my side, there is only one instruction
Yes, sir.
that please speak with the your gap analysis, what is the situation and result and the future prospects. That's 3 things that should be elaborate more. Nothing else. Okay, and please speak with the time best.
Yes, sir. Yes,
But
sir.
I think, madam, he was not clearly audible. Is it a problem at my end?
No, it must be at Nishad sir.
His voice is not clear at all.
Yes, ma'am. Same here.
Yeah, we could not hear you properly, Nishad.
Yes, now am I audible?
Still, it is not very clear.
What you do, Nishad, you are a little feeble. Your voice is feeble. You try to increase your volume. Yeah, that would be more. Or else, actually, if you want to save your bandwidth, you can put off your video. Then your voice will be clear.
Okay. Now is it clear? Hello?
Not yet.
Not yet.
Now?
Yeah, you can proceed, but you can even type and just put up the instructions. No, that can help us to proceed faster, right?
Okay. Okay.
Questioning will also have to be done in a loud voice, sir. Nishad sir.
Yeah.
Yes. Yes.
Yeah. Should we start then, sir? The Mankar sir and Nishad sir.
Yeah. To my end, no issue.
Yeah. So I now call upon Miss Aisha to present. Aisha Hazra. Aisha, are you there?
No, ma'am.
Okay. I will move on to the next participant, Ms. Nishigandha. Patil sir, can you confirm she is there?
Yes, ma'am.
Nishigandha is there.
Yes.
Nishigandha, kindly present.
Is my screen is visible?
It is about to be now. Yeah, it is visible.
Sure.
Continue, please.
Yes. Good morning, everyone. I'm Nishigandha Mahindra Shirsat from Kekewa College, and I'm delighted to present my research work in ethnomedicine, that is study of traditional plants in drug discovery, in this prestigious international conference. Me and my guide,
Your
assistant
voice is very
professor.
low.
Hello.
Nishigandha, kindly speak in loud voice, please.
Yes. Me and my guide, Assistant Professor Varsha Mistry ma'am, working on phytochemicals of traditional medicine.
Still, it is not audible clearly. Please adjust your audio properly.
From my side, it is 100 ma'am volume.
No, but we can't hear.
Nishigandha, what you do, you try to speak little loud and clear and be precise actually when you communicate properly. Okay?
Okay, okay.
Be little loud.
Okay, okay. So our work is in phytochemicals of traditional medicinal plant, Egel marmelis, which is well known as bell. Now it is okay.
Yeah.
Yeah,
Our
it's
aim,
okay.
our aim is to evaluate phytochemicals of Egel marmelis using in silico approaches to identify potential inhibitors of Salmonella adhesin proteins. Motive behind our study is salmonella infections, which is a major problem in worldwide. Also, the rise of antibiotic resistant species. As we all know, salmonella pathogenesis lead by adhesion to host cells, which is mediated by adhesin proteins, outer membrane proteins. If we block these adhesion proteins and process, we can prevent bacterial colonization and infection conveniently. So, reason behind choosing the bell is its phytochemicals are not yet screened against Salmonella adhesins. The phytochemicals of bells are showing rich activity in the pharmacological aspect, and they contribute in significant activities. We study on 18 phytochemicals and screen out them which show wide range of antimicrobial activity with less drawback. On other hand, there are various Salmonella OMPs that are outer membrane proteins, but some are actually responsible for provoking our immune response and involved in adhesion, invasion, biofilm formation, etc. The objective of our project or research is to evaluate the potential of Egel marmelis phytochemicals as inhibitors of Salmonella adhesins, to retrieve and prepare 3D structures of Salmonella, to collect and prepare phytochemicals of Egel marmelis, and to perform the molecular docking, analyze the binding affinities and interaction, and also to identify the top ranked phytochemicals with strong inhibitory potential against the adhesins. So, before moving towards the methodology, we perform the homology modeling using the Swiss model. As we are focusing on the OMPs, it is useful to understand their 3D structure, and it is helpful to understand the active site, identify the binding pockets of proteins, and provide a target models for molecular docking with ligands. Then we move towards our methodology. Methodology includes the protein preparation. The proteins are retrieved from the NCBI PDB. We model them, clean, minimize the energy to make the protein stable. Then we prepare the ligand and perform docking and visualize the binding affinity and interactions in the pi mole and interpret the data. These are some visuals of my research. First is the visualization of protein in pi mole, and this is molecular docking. Now, the interpretation of result is the analysis of phytochemicals, the affinity with adenosine proteins. As the ligands I have used, ligands are nothing but the phytochemicals which I have chosen and mentioned, that 18 phytochemicals. They are showing the highest binding affinity, that is greater than from -7 to 9. As we all know that from -7 to 9 kilocalories, if the compound showing binding affinity, then they can use for the further analysis for the drug discovery, like the ADMET and all. And here I also present my graphical representation data. In conclusion, I will mention out that in our research, the Eagle-Marmellus phytochemical shows strong binding affinities with all the selected Salmonella adenosine proteins. I also notice out the compounds such as agiline, flavin, imperotin, and oraptin, which showing the promising inhibitory potential, and also perform the docking, and the interaction suggests that we can use such phytochemicals in our further in vitro and in vivo research. So I will conclude on the note that nature still holds the solution of each and everything. We just have to driven our focus on them, and it is now easy by using such AI and in silico insights. So we can bridge the ancient science and modern science for best future. Thank you.
Judges, Dhananjay sir.
Yeah. No comments as such.
Nishad sir.
Okay, no comments from my side. Just one question that uh, have you performed any kind of ADME analysis using this phytocompound?
No, sir. At initial level, I perform the docking only, but in my further research work, I will perform ADMET.
Nicely done, Nishigandha. You can stop presenting now. All participants are requested to switch on their videos as soon as you join for presentation. I call upon Rutuja Hazare. Rutuja.
How many total participants are there, madam?
19, sir.
Okay. And are they going in the same sequence? Anyone
Yes, sir.
absent? Okay, fine.
No, like we come to know now only. So accordingly, I'm going according to the sequence. I am at IS 9 at present now. Rutuja is there. Ruchida, kindly unmute yourself. Rutuja. Okay, we'll get back to Rutuja later. I call upon Shubhadeep Datta. Rutuja,
Good morning,
you're
ma'am.
able to? Yeah.
Ma'am, I'm Rutuja Hazare, and there's a slight network issue here. I'll just fix up in a couple of minutes and I'll get back, ma'am.
Yes.
Thank you.
Shubhadeep Datta. Shubhadeep Datta. Yeah, thank you, beta. Please switch on your camera too.
Yes, ma'am. Am I audible?
Yes, you are.
Okay, ma'am.
Switch on the camera too.
Yes. Is it not showing my not showing my video?
Now it is. Yeah.
Oh, okay, ma'am.
Yeah, thank you.
Okay, good morning, everyone. Today, in nowadays, antimicrobial resistance has emerged as one of the most critical threats to the global public health. And among the resistant pathogens, Klebsiella pneumoniae is one of the most particular concern due to its ability to produce extended spectrum of beta-lactamines, such as SHB1 and CTXM. These are the enzymes that hydrolyze beta-lactamase antibiotics, rendering them their effectiveness and severely limiting their treatment options. Traditional approaches to combat resistance, such as developing new drugs, are often costly and time consuming. An alternative strategy lies in exploring the wild edible plants, which are the part of our traditional diets and ethomedicines. Such plant contains contains various phytochemicals, which has potential antimicrobial and enzyme inhibitory properties. Yet many remain unexplored, but our study focuses on the regional plants of Bengal, especially on L. samantosa. So this plant has a few compounds which shows promising antimicrobial activity and ESBL inhibition properties. So the main aim of our study is to find plants to isolate a few phytochemicals which show promising docking results against the antimicrobial against the ESBL proteins. So at first, I have worked with these two protein, SHG1 and CTXM. So the the methods that I used in this study, at first I isolated the protein, found the protein from PubChem database. Then I performed homology modeling. After that, I prepared the ligands and prepared the protein structure by cleaning and removing the water molecules and by adding charges in Autodoc Vina. After that, I performed docking in Pyrex. And then after visualizing Bioreva, I sorted out a few compounds depending on ADME analysis. So these are the few compounds which showed promising results in my study. 4-allyl 2,6 dimethylphenol glucopyronoside beta amarine and so on. So after that ADME screening, this shows these are the screening results. These three compounds showed the most promising results in my study. So scroside and 4-dimethylalanine glucopyronoside and irexoside, this showed the excellent ADME profile screening, and so that we can use them directly as an oral drug without much modifications. These are my docking results for SHB1 protein. Here, luteinol acetate and luteinol showed excellent docking results. First one having the docking affinity of -9.1 and the second one is 8.6. After that, CTXM, this showed this has lupinol acetate and iroxide D. These two compounds showed the most promising docking results. Next, these are the few protein complex which I try to visualize. The first one is SHB1 and luteinol complex, and second one is SHB protein with lupinol acetate. They showed excellent hydrogen and hydrophobic bondings. Next, complexes CTXM with lupinol acetate and CTXM irexoside D. Okay. So next, the main motive of my study is to isolate those compounds, and for further studies these compounds can be utilized because they show a promising affinity towards the ASBL protein. This can be utilized in used in ADMs later in vivo and in vitro methods to create a promisable drug which can cause less harm or no harm to our system. So after that, the this ethanobotany, my study specially highlights the ethanobotany of the highlights the part of the antimicrobial the ethanobotanical compounds which were used to deal with the antimicrobial resistance. First, Next one, thus, thank you everyone for listening it. By saying that, I want to conclude my session. Thank you.
Dr. Manka?
I want to understand from the participant that what is the scope of your study, making it more evident in terms of the antimicrobial resistance that you are trying to talk about. Could you please highlight on this?
Okay, sir. The first, if we try to deal with the antimicrobial resistance by creating new synthetic drugs, then it's going to be more costly. But if we try to find the possible solution from the local region plants in my study, I used plants from Bengal region, especially Ansamantosa. They show a promising docking results in this study. So from them, we can get easily bioavailable phytochemical compounds. And that can be easily used to formulate cheap drugs, which can be affordable for the lower class, economically lower class peoples.
Okay. Oh, fine.
Okay.
Yeah, it's fine for my end.
Thank you. Very well done. You can stop presenting.
Okay.
Now I call upon Neha Wagmare. Neha? Neha, kindly, yeah. Switch on your video.
Am I audible?
Yes, you are.
Yes. Yes, ma'am.
Start presenting, yeah.
Just my slide.
Yeah, it is
visible.
about to start. Yes, it is about to start. You switch on your video too.
Hello, ma'am.
Yeah, it is about to start. Switch on your video too. Neha, the slide is about to start.
Okay, I'm just.
You have network problems, it seems.
It's
your screen is just as a blank screen. We are not seeing anything yet.
Can proceed with the presentation?
Neha, we can't hear you too. Your voice is breaking. We are not able to hear you. Unmute yourself, beta.
Hello, ma'am.
Yeah, beta. Yes, Neha.
Okay, ma'am.
Yes.
We are able to hear you now. And see you too. Just start sharing.
Okay, ma'am.
You can switch off your camera. Start presenting.
PG student, department.
Neha, we are not able to see your presentation. You can switch off your video, but present. Start your slides. We are not able to see your slides.
Okay,
presentation.
Now, ma'am.
It has not started yet.
Hello.
It has not started yet. Neha, why don't we give you some time? You fix this issue and join us back.
Okay, ma'am. Okay.
Yeah. I request Aryan. Aryan?
Hello, ma'am. Can you hear
Yes,
me?
please. Yes, yes. Yes, beta. Please share your presentation.
Is my screen visible?
Not yet. It is about to. Yes, it is visible now. Please go ahead.
My presentation is visible, right?
Yes, yes.
Okay. So, good morning, everyone. My name is Aryan Ganukardekar, and I'm from the Department of Microbiology, Srivasadan, R.K.Talreja College. And today I'm going to be sharing with you of my study based on multi-target docking of phytochemicals against resistance-associated protein. So, phytochemicals are emerging as the potential drug targets in tackling antibiotic resistance. As we know, antibiotic resistance is a major threat, which many organisms like bacteria becoming multi-drug resistant due to the overuse of certain novel antibiotics, and there is only a certain aspect or limit of the novel antibiotics we can use currently. So, what do we do to tackle those challenges? So, the research is now being shifted more towards the plants. So, we can use the plant phytochemicals as they are to uh, due to their structural diversity and the already mentioned bioactivity in the Vedas or in our ancient sculpture. So now the reason more focused on these phytochemicals. Then what's the rationale and gap analysis? Phytochemicals have become topic of the research, and we are doing in silico study of this phytochemical or multi-target docking to reduce the wet lab experiments. So the aim of is to check the broad spectrum activity of the antibiotics against some resistant associated proteins which help bacteria to gain the resistance towards the antibiotics, which include ARC-AB, NOR-A, MEX-A, CDR1P, and TOC. So what are the resistant associated proteins? These proteins are part of the bacterial system which give bacteria its ability to become resistant to certain drugs. These proteins are usually efflux pumps or sometimes enzymes which inhibit or like throw out the enzyme or the antibiotic which is used to tackle those, and it results in superbugs and enzymes or some antibiotics which search are also not passing the clinical trials because they are not safe for use. So what do we do? These are the protein structures here for which we are going to see the docking study. Then this is the methodological workflow. First, we take the protein structuring source. We retrieve protein from RCSB PDB, which is the protein database. Then we, or if certain protein structures are not available on the RCSB PDB, which have been not widely studied. So we use homology modeling where we take NCBA sequences, faster sequence from the NCBA, and then construct a homology model using Swift modeling. Then we go for ligand curation, which we take ligand from PubChem and then go for molecular docking in Autodoc, Vina, and Pyrex. These three tools were used to perform multicellular docking. And the lastly, we produce ADMET study to see the antibiotic gives the phytochemical results if it is suitable for human use or not. This is the same in the flowsheet format. Then these are the results for some of the binding affinities we form from the phytochemicals which you see, ligand, then this 794 and 5770. These are some 3D interactions of the proteins and the ligand interaction. The part in yellow is the ligand and the rest of the protein interaction. This is the 2D structure, which here we can see in the MEXA, TOLC, CDR1P, and the NORA. We can see that there are stable covalent hydrogen bonds between the ligand and the proteins, which suggests that it could be used as a potential target. And these all are the same. These all 2D structures are of the same compound, which is camphor. Now, coming to the conclusion part, phytochemicals demonstrated significant binding affinities to the proteins. There were 42, for my study, I took 42 phytochemicals, which were screened accordingly and following the ADMET results and the docking scores. From the 42, we found 3 of the ligands of the phytochemicals namely 528683, camphorol, 5288074, stigmasterol, and 5770 rasparin. This showed the potential for binding all the mentioned proteins and have shown the potential as the broad spectrum antibiotics. But after the added screening, only camphorol was showing the good ADME results. And the reason why I have not mentioned camphorol here as a potential source, because its body absorption is very less. Camphorol is usually found in broccoli, tea, kale, and many day-to-day life foods and fruits. So it is easily available phytochemical. We should include those foods which contain camphorol to increase our immunity as such. And the results of this study at the end will say that we have found three ligands which are showing broad spectrum antibiotic activity. Camphorol is also showing good ADME results. So, but this this study can be used to further confirm or utilize to further narrow down the wet lab experiments. As we know, wet lab experiments are very costly and can also be time consuming. So if we have a previous base for these studies, the further research becomes easy. These are the reference. These are the future aspects for the chemical, why it is necessary, and these are the references. Thank you.
Dr. Mankar. Mankar, sir. Sir is not there. Nishad, sir, kindly go ahead and ask questions if you have.
I just have one suggestion, not a question. That in reverse working approach, what you will do, you will first find out the best phytochemicals or best. You have to cite the literature according to find out the best phytochemicals or base compounds, and then we have to perform the docking. Here, the ADME analysis is already performed by some another scientist or some other researcher. You just have to dock it against various proteins. Okay, so in your study, there is a no, we can say we can skip the ADME part. That's it. So much. This is the suggestion.
Okay, sir.
Thank you, Aryan. Just let me check once again if Dr. Mankar is available. Mankar, sir. Ranjana, ma'am, kindly find out. We can go ahead or should we wait? Aryan, thank you so much, Beta.
Thank you, ma'am.
Okay, ma'am, Manikar sir has messaged me that please proceed from questions from your side only. We will inform. So,
Sorry?
please, Manikar sir is
Nishad, sir,
saying,
we can't hear you very well.
okay, now am I audible now? Hello?
You are audible, but it is not clear.
Okay, Manikar sir is saying that please proceed from questions your side and inform them according to this.
Yeah, okay. So I call upon Vaishali now, Miss Vaishali. Vaishali Salve.
Yeah, 1 minute, ma'am. I'm giving her permission.
Yeah.
Yeah, Vaishali, now you can start.
Yes, ma'am. Good morning, ma'am. Good morning, all. Let me
Switch on your camera first, Beta. Unmute yourself. Yes, very good. Now present.
Ma'am, is my screen is visible?
It is about to now. Just give it. Yeah, it is visible now. Put it into slideshow.
Yeah.
Above. Okay. Yeah, it is there. Now you can start.
Good morning, all. This is Vaishali Salve from Institute Degree College. My today's topic is actually about the Computational analysis of small molecule interaction with Hall A. Hall is DNA polymerase DNA polymerase III का delta subunit. So basically, my aim is to explore the potential of small molecule inhibitor जो कि target करेगा
Vaishali,
delta subunit.
your voice
Yes,
is not very clear. Your voice is not very clear.
hello. Ma'am, now
Yeah.
I am audible.
Yeah, you are audible, but it is not clear.
Okay, ma'am.
Try speaking, unmute yourself. All participants are requested to check out the availability of uninterrupted network. Sit in an area where you can provide us with a continuous flow of network and your presentation. Vaishali, do you need to log out so I can take the next participant in the meantime? Vaishali, kindly respond. Unmute yourself. Patil sir, I think she is having difficulties in network.
Okay, ma'am.
Can we stop her presentation for the time being and take the next participant in the meantime?
Okay, okay, ma'am.
Yeah, she is there, I think. Just give her a minute.
Hello, ma'am. I am audible.
Yes, yes, you are. Please go ahead now.
Just a minute, ma'am. I will join again. Actually, I am not able to present.
You are able to. We can see your slides. Patil sir, I'll call the next participant in the meantime.
Yeah, yeah.
Yeah. Basudha, please join till Vaishali gets ready. Basudha Laha, are you there? Yeah.
Yes.
Yes,
Yes,
please.
ma'am. Am I audible?
Yes, you are, beta.
Okay, ma'am. Ma'am, am I?
I can see you, but
Ma'am?
your presentation is not yet seen.
Yes, I'm not able to share my screen. I don't know why.
What is the problem? Is it showing something?
Yes, ma'am. Wait. There is a share option, but it's not getting clicked.
Share option is not getting clicked. Okay, why don't you take your time? I will take on the next participant in the meantime. You figure it out. Let me know as soon as you're ready.
Basudha, you need to check a window. Okay, when you click on share your screen, okay, either you
Yes,
need
sure.
to choose the entire screen or the application window.
Yes.
Once you click that, then click on share.
In Windows section also I can't share.
Once you click on the entire screen, does it highlighting the share option, or it's still showing?
No.
No.
It's
Okay.
still showing.
Then please log out and log in again. Okay, that may help.
Yeah, you try it. In the meantime, I'll call upon Anam. Mohammad Anam Shafiq Ahmed Ansari. Are you there?
No, ma'am.
Okay. Neha Wagmare, you are ready?
Yes, ma'am.
Yes, please. Go ahead.
Okay, ma'am. Hello, ma'am.
Yes, beta, your slide is
Am I audible?
visible. Yeah, audible as well as slides are there. Please start.
Okay. So hello everyone, myself Neha Sunil Bagmare. I'm a PG student, Department of Microbiology, R.K.Talreja College, Mumbai University. The title of my project is a modeling of essential proteins of Mpox virus and doc known broad spectrum antiviral compounds to explore target ability. So let's start with the introduction. So what is the Mpox? So Mpox is a viral illness caused by the monkeypox virus, and it's a membrane of orthopox virus genome, and it's also included in the virus which is responsible for the smallpox and cowpox. It was first identified in the monkeys in 1958, and the first human case was reported in the Central Africa in 1970. It was spread through the close contact with the infected animals, humans, or contaminated materials. I choose these proteins for my projects: VP37, VP39, D13, 17L, TMPK, which is thymidylate kinase, F8, because these proteins are essential for the different stages of viral life cycles, replication, assembly, and release. So they are considered maybe a promising protein targets for Mpox virus, and I used this antiviral broad spectrum compound against them: tecovirimat, pseudofever, curcumin, ribavirin, valacyclovir, trabivirin. This compound I used against them. So rational and gap analysis. So there are only a few antivirals drug explored and available against the Mpox, and some of them not fully explored, and the lack of data. Many of the essential proteins of Mpox structure are not experimentally present in the PDB. So these are the main important problem. So that's why the underexplored drug target interaction, the broad spectrum antivirals are widely used, but their specific binding interaction and mechanism toward Mpox are not fully investigated, fully explored. So bioinformatic advantage is that in silico protein modeling and docking can rapidly predict the target ability and prioritize the compounds for the experimental validation. So my aim of my project to model the essential Mpox virus, because these structures are not present in the PDB database. So model the Mpox virus proteins and identify the potential antiviral compounds. And the objective is to select the essential viral proteins, perform the structure modeling, dock the broad spectrum antivirus, and evaluate the binding interaction for this therapeutic potential for to explore target ability, which we can use in the future. These are the methodology I follow for my whole research. So the lack of the data in a PDB database, so I retrieve the protein sequence from the NCBI and predict the protein structures using a homology modeling in a Swiss model. Then the protein preparations are done in a PyMol. In PyMol, I just remove the water molecules, unwanted chains and ligands, so it does not interfere when I performing the molecular docking with my antiviral compounds, and apply the charges in autodock vena, so the structure can balanced. My protein was ready for the docking. For the ligands, my antiviral compounds, I retrieve them from the PubChem. I check this information regarding if they are useful for humans. They are not harmful for any. They do not show any harmful significant symptoms For the humans, so I use the Swiss adenine. Then my ligand preparation is done in the open Babel, and my ligand is ready for the docking. Then the molecular docking is done in a Pyrex. So after the docking, I got the result in Excel sheet, in Excel sheet data. So the last step, we can just visualize in this in a bio discovery studio, where we can get a 3D and 2D interaction of these proteins and ligands. These are my protein. I predict the structure using a homology modeling in a Swiss model, VP37, VP39, F8, D13, TMPK, thymidlase kinase. These are the result I got after the molecular docking. I dock every protein with every ligands, and I just choose the best result showed. So here the take covariant shows the best result, strong binding affinity to all my 5 target proteins. So these are the 3D and 2D interaction I got from the BioWare Discovery Studio. So these are the 2D and 3D interaction. VP37 with a tecovirimet antiviral drugs, then the VP39 with the tecovirimet, the F8 with the tecovirimet, the F8 with the curcumin, the F8 with 17L with the tecovirimet, and the D13 with the curcumin, then the D13 with the tecovirimet. So all the analyzing the result I got after these molecular docking, I was successfully modeled the 5 essential proteins, which is 17L, D13s, because these data are not present in a PDB. So I successfully modeled them using the homology modeling. Then the molecular docking showed the tecovirimet as an existing antiviral drug. It's a strong binding affinity to all my target proteins. So it is and it is a potent broad spectrum antiviral drug for mpox treatment. And many of the researchers are used them in these in their research. Then the compound curcumin also showed a strong binding affinity towards some protein. So it indicating that it could be a promising new lead for developing antivirus, anti-mpox drugs we can use in future. Our analysis confirmed that these proteins which I choose select for the project are a promising target, and we can use them in developing a new antiviral therapies drugs against them. Then the computational studies serve as a crucial step, paving the way for the further in vitro and in vivo studies to validate these findings and accelerate the drug discovery process. The future scope. So these studies should be validate in vitro and in vivo studies to confirm the efficiency and safety of the antiviral compound against the mpox. So it is safe for the human use, and the result there shows. Then the multi-target approach can be studied. Can explore the simultaneous targeting of multiple viral proteins to prevent the resistance and improve the treatment. The repurposing may offer faster treatment options, but there are some we have to or uh, we have to some research on that. Then the advanced simulation can improve the understanding of interactions. Then the AI tool can use to screen more compound efficiently. These are the references I use for my whole research. And thank you.
Nishad sir.
Yes, no comments from my side. Just one glitch is there. I could not see slide prepared by. So can you please
Sorry
send it
sir.
to me? Uh, I could not see the slides prepared by her. So
Okay.
can send me in the chat
We will
afterwards.
check that out, sir. If you do not have any questions, Neha, thank you so much. Done very well.
Thank you, ma'am. Thank you, sir.
Yeah. I call upon Rutuja to join now. Rutuja.
Meena ma'am, can we hold it for 5 minutes?
You want to? Okay. Rutuja,
Yeah,
just
that,
wait for a
let
few
that
minutes.
other judge join. She's joining at 12.
Yeah. Rutuja, kindly hold on.
Okay, ma'am.
We will just take 5 minutes for the judge to join. Bharti ma'am, welcome. Friends, our judge has just joined, Dr. Bharti Wadekar. I take this opportunity to introduce Madam. Ranjana ma'am, is it okay? I'll go ahead.
Yeah, ma'am, please. Otherwise, okay. If it is okay with you, please go ahead.
Yeah. Dr. Bharti Gade, sorry, I'm so sorry, Gude Wadekar is an accomplished faculty, microbiology, interdisciplinary department at ZSCT's Thakur Shamnarayan Degree College, Mumbai. She has served as IQAC coordinator there. She is a NAC advisory for 4 institutes. She also has done research on marine microorganisms with probiotic properties, particularly bacteriocins production and its application as antimicrobial agents and food biopreservatives. Her doctoral work, supported by DST-WOS A fellowship, explored the antibacterial activity of bacteriocin from marine probiotics and structural analysis by bioinformatics, with a project valued at rupees 17,74,000. With over 14 years of professional experience, Dr. Wadekar has held teaching and research positions across prestigious institutions in Maharashtra. She has published extensively, including 10 full-length papers, 10 book chapters, 9 books, and numerous conference proceedings, alongside securing 6 patents, 3 of which are granted in Germany. Her work spans bacteriocin production, biosurfactants, and bioinformatics, with notable contribution to environmental and health sciences. She has presented at international conferences and received numerous accolades, including the Best Researcher Award 2022, Global Youth Icon Award 2022, and multiple certificates of excellence in peer review. Her contribution extend to academia through engagement as guest speakers and professional reviewers. Ma'am, we are very proud to have with us today. Thank you so much for accepting at the shortest of notice and being sweet enough to accept. Welcome, ma'am.
Thank
So
you, ma'am,
if
for the opportunity.
If the judges are ready, may I call upon the participant? Judges,
Yes,
is this
ma'am.
okay? Yeah. So I call upon
Yes,
Rutuja.
yes.
Thank you, sir. Rutuja?
Yes, ma'am.
Yes, please.
Ma'am, I'll start sharing the screen.
Not yet. You have not shared. You switch on your video also.
Yes, ma'am.
There is a lot of background noise from your side, Rutuja.
Ma'am, can you see my screen?
Yes, I can see your screen. Put it into slideshow mode.
Yes,
Switch
ma'am.
on your camera. Slideshow will go on the top. There you can see.
Ma'am, can I start?
You can start. We are not able to see you.
Ma'am, I have a slight problem with my camera, but I've on it, ma'am.
Okay. Put your slides in slideshow mode.
Yes, ma'am. Good afternoon, everyone. So my topic is in silico screening of medicinal plants derived from plant-derived phytochemicals as potential inhibitor of breast cancer proteins. Basically,
Rutuja, your slides are still not in slideshow mode, beta. There's a lot of interference, background noise. Do you have earphones?
Ma'am, can you see my screen now?
There are multiple screens at present.
Yes, yes, go to the slideshow.
Yeah, yes, sir. Can you see it now, ma'am?
Yes, yes, we can. Please start.
Cancer. So the uncontrolled cell proliferation caused by accumulated genetics and epigenetic alteration that degrade the growth, apoptosis occurs. So basically, by the genome stability and microenvironment treatments, which includes the surgery, radiotherapy, and hormone therapy. There are such therapies for the cancer treatment. Specifically, I have chosen the breast cancer. In that, okay, so breast cancer, it is a malignant tumor which is found in lobules of milk producing glands. Basically, this is very much most common in women worldwide, and that can lead to the cancer death. Early detection can be improved the outcomes. So for my project, uh, sorry, for the breast cancer, the risk factors may be genetical, maybe hormonal, maybe because of the lifestyle. Genetical, there may be mutation occurring in the genetics. Hormonal, there can be excessive exposure of the estrogen and all, which leads to the breast cancer. And because of the lifestyles, also lack of exercise, more consumption of alcohol can lead to the breast cancer in women. Next, the proteins leading breast cancer. We have there are many proteins which can lead to the breast cancer, and from that we have selected the most two important, that is HER2, which is the human epidermal growth factor, and the second one is BCL2. Um, to treat the breast cancer, we are taking our medicinal plants as the inhibitors. We have selected the three medicinal plants, that is Andrographis paniculata, Calotropis gigantea, and Sphagneticula calendicia. So these plants, from these plants, we shortlisted many of these phytochemicals um, under selections like Libinski rule of five and other things. And after shortlisting that, we we selected the phytochemicals, and we binded them with our proteins, selected proteins, which after binding had showed the as far the better results. So the materials we required was the cancerous proteins, medicinal plants, and the AI tools, from which we were going to, we did the auto docking. So we started our project by very basics, by searching the proteins and then the ligands, and after that we shortlisted the proteins and ligands which can be which can inhibit and can cause the can.
Rutuja, kindly continue.
Can you hear me, ma'am?
Now we can.
Okay. So after that, after docking certain phytochemicals with the proteins, we got this result, which were so far better. And from this, we got more than 6 to 7 phytochemicals which had the most binding affinity towards our first protein, which we did. And the process is still going on, ma'am. We have the second protein also, and we'll do the same process with the second proteins. And after the selecting the most highly binding affinity, we can take these phytochemicals as our therapeutical medicines in future predictions and future strategies for the treating of breast cancer.
Judges,
Thank you.
Dr. Bharati, Dr. Mankar, and Nishad sir.
Yes, ma'am. Uh, can you tell me the which software is used for the molecular docking?
Rituja, you have to answer to the questions. Unmute yourself.
Yes, ma'am. Can you please repeat the question once again, ma'am?
Uh, in a flow sheet, you already shown the molecular docking. So can you tell me the which software used for the molecular docking?
Ma'am, we use the Autodoc, ma'am.
Okay. And one more thing now, why do you choose या फिर check the ADMET properties?
Ma'am, we did that for the Lipinski rule of five, which has the certain five rules under which we can see the molecules are with zero violations and no violation, no Lipinski. If there is more than two or more violations, we can't take that phytochemicals, ma'am. They can't be used as a drug in future, ma'am. If it is zero or at least one, we can use it as a drug in future. They can inhibit, they can regulate it.
Okay. Now tell me the which ligand and the which protein you choose for the interaction between.
Protein, the first protein we did docking was the HER2.
HER2 and ligand.
Ligands are the phytochemicals of the three medicinal plants, ma'am.
So every ligand you used.
Yeah, ma'am. We shortlisted the phytochemicals. Then, by the smiles, by taking the smiles and going to the Swiss addendum, we shortlisted which has only zero or no violations, ma'am, which comes under Lipinski rules.
And then whenever the result obtained, you validate that the result with any other software.
No, ma'am.
Okay. Okay. ठीक है.
So
Dr.
next,
Mankar.
do with the same process with our second protein also, and then we are going to see the results.
Okay.
Yeah.
For the future predictions.
Okay.
Dr. Mankar, do you have any questions, sir? Mr. Nishad, please go ahead with your questions.
No, ma'am, I don't have any question. We can.
Yeah. Rituja, thank you so much.
Thank you so much, ma'am.
Yeah. Now I call upon Aishi. Aishi, you ready? Aishi Hazra.
Wait, madam. Yes, Aishi.
Aishi, unmute yourself. Aishi Hazra.
Yes, ma'am. Am I audible?
Yeah, you're audible now. Start your video as well as presentation. Put into slideshow.
Ma'am, is it visible?
It is visible, but put into slideshow, beta.
Ma'am, I've put it into slideshow, but
Okay, do one thing, come down here. Can you see glass, our glass? Next to your 100, yeah, 100%. Come to the left. The R glass, R glass. Yeah. Not that. Is it that? Not that. The R glass. Yeah.
Ma'am, is it visible?
It's still not into slideshow mode. Just see to it that you keep on scrolling the slides then. Okay. Let it go. You can start the presentation. Go on scrolling the slides, but
Okay, ma'am.
start.
Good afternoon, everyone. My name is Ulshi Hajra and I'm from Department of Microbiology, St. Xavier's College, Kolkata. Today I'll be presenting my topic on host pathogen interaction modeling of human inflammatory receptors with fungal and viral effector proteins. This work basically focuses on understanding how microbial effectors target host immune receptors and how computational modeling can reveal their impact on immune signaling pathways. Um, so, host pathogen interactions are very dynamic processes. Pathogens produce effector proteins that can hijack and manipulate the host cell machinery. Key inflammatory receptors such as toll-like receptors, caspase-8, FAT, and certain cytokines receptors act as the first line of defense. But fungal and viral pathogens often secrete some toxic proteins which disrupt and suppress these receptor-mediated pathways. So to study this, I used in silico approaches such as structural docking, um, uh, 3D interaction analysis, and signaling network modeling to predict how this effector and receptor interactions shape, to the are shaping the fungal infections. So this is my aim. The central aim is to model and characterize the molecular interactions between host inflammatory receptors and certain selected viral effectors, as well as fungal, to evaluate their impact on immune signaling and pathogens. So the specific object include for caspase-8 with E6 and E7 viral proteins to predict whether whether these proteins modulate apoptosis to promote immune invasion or not. Then FAD with E6 and E7 to study potential disruption of death-inducing complexes, which is crucial for apoptosis. And then IL-1 R1 receptor with a fungal toxin called candidialysin to investigate its effect on this receptor, which is responsible for inflammatory signaling. So my work, like I used this workflow to study this interaction. I retrieved the proteins from PTB and that protein data bank and Uniplot, and then there were certain structures which were not available, so I modeled them using the Swiss model, and then I prepared the proteins like removing the waters, adding charges in PyMOS and PyRX, and then I did the docking of the protein-protein interaction in HPEP-doc, and finally visualized the interactions using BioWare Discovery Studio Visualizer. So these are the proteins. This is ALS, cadhesin type of protein from Candida. This is also a fungal toxin protein from Candida. This caspase and FD are the proteins which are responsible for inflammation. Then E6 and E7 act as viral oncoproteins, which are derived, which are present in HPV 16 and 18. And this particular receptor is responsible for inflammatory responses in humans. So these are the results of the docking score and the binding affinity of the interactions. The affinity energy was quite favorable. These are my the 3D interactions. So in conclusion, I draw that caspase in and FAD targeted various viral proteins of E6 and E7, and this IL1R1 interacted with the fungal effectors and toxins, candidolysine and ALS3 proteins. Uh, these biological implications are significant, which can help us to identify or to uh, modulate drugs accordingly, because they show this effectors showed very much good binding affinity with the receptors, and hence can provide a mechanistic understanding of host pathogen molecular crosstalk as well as therapeutic opportunities to restore immune balance and combat infections. These are my references. Thank you.
Yeah. Dr. Bharti?
Am I audible?
Yes, ma'am.
Okay. Can you show me the slide number 8?
Yes, ma'am.
Yeah.
Yes.
Please give me the first 3D structure interaction explanation. Yes.
Ma'am, sorry, I could not hear you.
I'm not audible.
You are,
No, I
ma'am.
could not hear your question.
Okay. See, first 3D interaction of your ligand and the protein, whatever. Yes. So can you explain that diagram?
Uh, Ma'am, this diagram basically shows the interaction between E7 and fat protein. So basically, this is showing the all the favorable bondings, including hydrogen bonds with the ligand, not ligand, with the effector with the protein, like the which amino acid is being bonded to which amino acid in the protein.
Okay, fine. Uh, can you tell me the what are the challenges in a host and a pathogen interaction modeling?
Challenges include, firstly, the host. If I'm considering the host, the proteins are very big, which are difficult to model, and the effectors, um, sometimes the effector can have some mutations, which will serve as a challenge when I'm studying this kind of interactions.
Okay, you use the software HADDDOCK, right?
Yes, ma'am. HPEP.
Yes. So is it online available?
Yeah, it's a web tool.
Okay, fine. Thank you.
Thank you, ma'am.
Thank you, Aisha. You can stop presenting. Now I call upon Vaishali Netkar.
my slide is visible or not?
Yeah, slide is visible. Presentation mode is there. Please go ahead.
Thank you, ma'am.
Speak a bit loudly.
Yes, ma'am. Good afternoon, everyone. Today I will present my study topic on urease regulation proteins, bacillus in bacillus. So first of all, urease is a nickel-dependent enzyme which plays crucial role in a nitrogen metabolism. So these activities is tightly controlled by the accessory proteins which are URED, UREF, and UREG, which plays crucial role in the activations and maturations of urease enzyme. If this if these accessory proteins get affected or altered or disrupted, it directly affects on the bacterial survival and its pathogenicity. So modeling is the modeling is the modeling of proteins, and it gives informations about the ligand interactions binding with uh, which gives the proper which gives the inhibitory targets to regulate or inhibit the enzymes, urease enzyme. So most studies focus on the urease direct inhibiting binding sites, but less study or less focused or on the indirect accessory proteins, which are URED, UREF, and UREG. Because of that, we have low informations about the structural and ligand binding informations. Because of that, we face the different challenges for finding the or design the target inhibitors. The computational modeling and docking approaches offers a bridge this gap, and we can and allowing us to predict the structures and test potential regulatory ligands for virtually. The main aim of the study is to identify the potential of interactions between proteins and ligands in bacillus, to explore the novel ligands with proper strong binding potentials, to explore the indirect inhibition strategies, to target directly target on the accessories proteins, indirectly target on the accessories proteins, not than the actual catalytic sites, to provide insights may guide antimicrobial approaches development in dip, in against urease dependent bacteria. So our flow starts with a literature review and collection of ligands. Followed that, format conversion is using by Open Babel, which converts the SDVF file to PDBQT file. After that, protein sequence is retrieved from the NCBI and followed by modeled by using Swiss modeling. The molecular docking is performed by using Autodock Vina Wizard via PyRX. This docking score is analyzed by Python and Excel sheet. The evaluate the lead likeness and drug likeness. ADME analysis is used, and the last one, the interactions between ligands and proteins, will visualized in the BioVia Discovery Studio. By this systematic approaches, we can assuredly uh, accuracy and reproducibility of the results. And this table uh, this table highlights the binding affinity of ligands towards the accessory proteins or macromolecules. Different interactions shows different potency or strong binding energies. So there it includes URED, UREF, and UREG. In URED, there are two macromolecules are selected, and in UREF, I selected 4 macromolecules, and in UREG, I'm selected 8 macromolecules. So these 3D structures indicate, you can see here the ligands fit within the protein binding sites, which are spectrally orientations of hydrogen bonds and hydrophobic contacts demonstrate stable docking poses. In this 2D diagrams, already structure shows the specific amino acids residues which are binding to the ligands. These insights are important for identifications and conserved residues that serves as a future drug targets. And finally, I'm here to conclude. So to summarize, computational modeling of URED, UREF, and UREG provided
reliable structures for model docking. Ligand interactions study reveals the compounds which can easily or we can maybe use for as a inhibitor for the urease maturations. So targeting accessory proteins as offers to indirect but effective strategies to reduce urease activity or maturations, which which could ultimately weaken bacterial survival and pathogenicity. These studies, these studies emphasize the value of in silico approaches in early drug discovery, and and though though experimental validations, which are molecular dynamics, biochemical assays, and in vitro studies, will be essential for further studies. And the last, I would like to acknowledge some reference papers or some references which supports my study. And thank you all for your attention. That's it.
Dr. Mankar.
Yeah, I would like to know from the researcher that yes.
Sir, I can't hear you.
Can you just put up your objectives, please?
Okay.
I'm not able to see your slide. Objective of your study.
Yeah, she's putting it into presentation mode, sir.
Yeah, one.
Shall put into presentation mode?
Okay.
Yeah, you can state it also. Now, if you see your objectives particularly, yeah, let me just go into it. So you are talking about providing insights for developing an antimicrobial approaches. Well, I am just seeing the two objectives which are more giving us some or the other level of finding. But antimicrobial approaches, what do you mean by antimicrobial approaches?
Okay. In ligands, I'm selected some inhibitory components which are inhibits the regulation site or accessory proteins which bind they binds to that accessory proteins and get inhibit or affects on the maturation of enzymes which produce urease.
Because I was little strange to state this insights, because insights is something, no, it's very subjectively actually we have to provide insights where like your study is more towards quantifying these things and you are getting a very solid information, you know. So we try to use a word which is more towards quantitative approach actually, right? Insights are one, like it's very subjectively. So I was little inquisitive about it. Okay, no ahead, nothing. Please go ahead.
Okay, sir.
Dr. Bharti.
Okay. You use the URD, URF, and the URG, right?
Yes, ma'am.
Okay. Out of that, which one is the suitable?
Most suitable is UREG.
UREG. Why?
Because they give high drug likeness and lead likeness towards the inhibitory components, the interactions between the ligands and the macromolecules.
And how do you evaluate your molecular docking result?
Evaluated from the PyRX, the CVS file, which converts it to the Excel sheet.
Okay. Okay. Thank you.
Yeah. Thank you, ma'am. Thank you, Vaishali.
Thank you, ma'am.
Now I call upon Miss Vaishali Salve. Vaishali Salve. Kindly unmute yourself, Vaishali.
Hello, ma'am. Yes, ma'am.
Yes, yes.
Ma'am, I am audible now.
You are audible. Kindly start your video as well as presentation. Yeah.
Yes, ma'am. Just a minute, I will share my screen. Is my screen is visible?
Screen is visible. Put into slideshow. Yeah, it is visible. Can start.
Yeah. Good afternoon, everyone. This is Vaishali Save from SSC Degree College. My today's work is at today's research is about the computational analysis of small molecule interaction with Hole. So now everyone knows that DNA polymerase are the enzyme which is used for generally used for the replication. And the main thing is that in bacteria, जो DNA polymerase है, it is a main replicating enzyme. And nowadays we have only target जो भी antibiotics we have made, yeah, we have discovered that only target to DNA gyrase, then topoisomerase, this kind of enzymes. But my study is related to the main root of that particular enzyme. As we know that DNA polymerase 3 is made up of lots of subunits. This is the structure of DNA polymerase enzyme, as it is having a lots of enzyme. And actually DNA polymerase 3 subunit is made up of lots of accessory subunit. So in that I have focuses on the delta subunit of DNA polymerase. Actually it is the subunit which help the clamp, that is a main processive enzyme जिसको वो help करता है. And if we target to that particular subunit, then we can target our inhibitory effect on that. So if you talk about the bacterial, actually this replication enzyme, it is absent in humans, because if you go toward why this is a drug target, because it is having a distinct active site. First is in bacteria, it is having a distinct actually DNA polymerase mein hi there is a thing, we are getting a distinct active site. Second thing is we are getting a larger finger domains. And very important is third target is our unique PHP type exonucleus domain, which is only found in bacteria, not in humans. Actually this is the histidine wala thing. In bacteria it is a synthesis pathway is there, but in humans this histidine is essential amino acid, so we don't synthesize that. So it will be a good target for that. Second thing, as I have talked about, we have only moved toward the DNA gyrase, then DNA topoisomerase, these kinds of enzyme. So my study will be a novel and untapped antibiotic target. Second thing is under explore drug target. Now next thing is the aim and objective. So as my study aim is to explore the potential small chemical inhibitor, small molecule जो कि target करेंगे the basically delta subunit of DNA polymerase enzyme. And this will be followed by the different steps, like screen of first screening the chemical compounds which will be suitable for my study. Then I will do the performing the docking. Then we'll see the protein ligand interaction with the discovery audio visualizer. And then we'll see the ADMA analysis with the help of a Swiss model. So basically the basic step is the identification and then validation. So first we have to novel target. First we have to see the what are the essential genes. So first we have target hall A. Then what kind of things ligand. Second thing will be heat identification. Then third one is your lead identification and then optimization. And fourth one is further it will be future, that is experimental validation. And fifth one will be a preclinical and clinical development. So this is the basic steps happen in drug discovery processes. Now the structure which is showing Hall A chain, which is our target, which will that is Hall A protein from E. coli. The very first step is as ligand preparation. So this has done with the help of a PyMol. I have downloaded the structure by minimizing the by removing the hydrogen bonds and different heteroatoms, and only I have keeping the whole HN. The second thing is your ligand selection. So for ligand selection, PubChem databases I have use, and in that I have selected lots of molecules. Basically, actually, 10 molecules I have taken. Some of were nucleoside analogs. Then there are some benzamide analog.
Then even I have taken the cepromycin. So from that, I could see, I will show you the binding. I have just taken some the binding affinity, which is giving me a high value, that is -9. Actually, it is a low value showing the highest binding affinity. So these two compound I have got, but one when I have checked for the first compound number one was giving, when I have done the adenine analysis, so it is given good effect. But this second compound has given me some lots of violation. Second thing is a grip preparation using a Pyrex. So for that, before that, we have converted PDPQ into, we have added the charges, minimizing the charges. This is the grid preparation. This is the binding affinity I have got. And now this is the thing, Discovery Studio Visualizer. I have, this is the 3D image where target and ligand interaction we can show with the help of some hydrogen bonding, hydrophobic interaction. If we see to a 2D image of these, so we can see of my ligand. So we can see that glycine is then cysteine,
Vaishali,
adenine.
you'll
These
have
are
to
the
come to
some,
conclusion now. Vaishali,
yes,
come to conclusion
amino
now.
acid. Yes, ma'am. This is the basic Swiss ADME analysis. So the compound one, which is I am talking about, it was the GI absorption is very high. Then bioavailability score was 0.55. It is a favorable drug life, but only in Lipinski's rule, it is showing only one violation because its molecular weight was 520. The next thing is, conclusion is, yes, this delta subunit can be a promising antibacterial target, and the compound one can be a strong lead candidate, as it is showing strong binding affinity, even favorable ADME analysis. Even my impact of my study is this: targeting of whole A subunit of DNA polymerase can offer a novel selective antimicrobial strategy to fight against our antibiotic resistance. So these are the some future direction. By doing this some experimental validation, we
Conclude
can
now, please.
further. Okay, these are the references, and I would like to acknowledge Dr. Anjana ma'am and Isha sir for their guidance and support throughout this my project. Thank you so much, everyone.
Judges, this is Vaishali's survey. She comes under the faculty category. Dr. Mankar.
Yeah, I appreciate the students' effort. She has done an exemplary work. I just want to know from the presenter that she's talking about computational results. So, Vaishali, can you tell me that when you are validating with experimental data, does it confirms the efficacy and safety of this?
No, sir. For that, we have to do this some clinical trials. First, we have to do the biochemical assay and all, cell viability assay. Then we have to do the clinical trial, and that we can do the safety analysis.
Okay. Next, I want to ask, what is the way forward to your study? Are you looking ahead to take ahead this study? What will be the next plan for your study?
Yes, sir. Actually, I would like to say thank to Ranjana ma'am and Nishat sir also. Actually, the theory we have learned during the academics, and when we do the practical, it was, अरे ऐसा भी होता है. So like the thing I have discovered, so it is very good thing to go towards research as well. As our antibiotic resistance is creating a global problem, health problem.
Yeah, good, very nice. Thank you.
Thank
Dr.
you, sir.
Bharti.
No question, ma'am. Nice work. Really nice work.
Thank you, ma'am.
Thank you, Vaishali.
Thank you, ma'am.
I next call upon Ms. Basudha. Stop presenting, Vaishali. You can switch off your camera. Ms. Basudha. Vaishali, stop sharing. Patil sir, can you help her out? Yeah. Basudha, yes. Vasudha, you can start. Yeah.
Ma'am.
Yeah, your screen is about to start.
Is visible.
Your screen is about to be visible. Your voice is cracking, but
Hello, ma'am. Am I audible?
you are, but your voice is cracking. You
Hello.
switch off your. Yeah, hello. Vasudha, your voice is cracking.
Yes, ma'am. Yes.
Your voice,
Yes.
audio is cracking. Not really heard. Breaking. It is breaking. Switch on your audio.
Now am I audible?
Yeah. Put your slide in slideshow mode. Start, please.
Okay, so good afternoon. My and I am a student of BSc Microbiology. Today, my topic is about comparative docking study of post-defense peptides against the surface adhesions of K. pneumonia and S. auris. So, introduction. As we know, in today's world, the antibiotic resistance has become one of the most pressing challenge. So, we can replace the antibiotic as the host defense peptide, also known as antimicrobial peptide. So, aim of my study is finding the antimicrobial peptide that can dock the protein or adhesive protein of Klebsiella pneumonia and also Staphylococcus aureus. First, we have to know the surface adhesin. Surface adhesin in Klebsiella pneumonia that I am going to work on is type 1 Fim-H adhesin. And in Staphylococcus aureus, I am going to work on the type 3 fimbriae, also MRK protein and MRKT adhesin. So what is AMP molecules? AMP is a short peptide chain that is containing 10 to 60 amino acids, and this can be found in our body also as a immunity. Types of AMP. We can find in mammals and microorganisms and amphibian and insect also. So there is the types of AMP cathelicidin and gramicidin, secropin. Methodology of the docking. So first I reviewed the literature and then select the AMP, then collect the sequence of AMP from a AMP database, then dock them into HPEP dock. And also I retrieve the sequence of protein, females and SPA protein, that is a Staphylococcus adhesin protein from the NCBI, and then I use a homology modeling, Swiss model, and then I sequence in HPP doc. After the docking, I visualize also them in HPP doc. These are my female protein and lactoferricin AMP docking visualization. This is the SPA protein docking visualization. So the conclusion. In conclusion, we can say we can replace the antibiotic with HDP, host defense peptide. So I think we can block the bacteria to form biofilm with the help of AMP molecules. Thank you.
Dr. Mankar.
Vasudha, you have raised a question on replacing the conventional antibiotics versus to this HDPs, right?
Dr. Mankar and the other judges, Let me know that Basudha is from UG category. Thank you.
Yes, sir.
Dr. Manka? Are you presenting, Basudha?
Hello, am I audible?
Yeah, you're audible, sir. Now.
Yeah, you were saying something, madam, about Basudha.
Yeah, that she is a UG category. I'm telling the category. She belongs to the
Okay.
UG category.
Okay. Yeah, fine.
I think she is not able to present. Is it Vasudha? You can answer. Vasudha, are you there?
Yes, ma'am. Yes. Yes, ma'am.
Okay, it's a very general question.
Hello?
Yeah. Am I audible to you, Basudha?
Yes, sir. Yes, sir.
Okay. You are just making a dialogue that we are trying more towards replacing this HDPs to the conventional antibiotics. So, how do we see that it is possible in terms of we find conventional antibiotics very affordable and the cost of this HDP is very high. So, is there any mechanism like you can see like when we are trying to project, because these studies will be projecting more, stating that now HDPs is the future because we find antimicrobial resistance a lot and all this going on. So, is there anything on this part also, like clinical development? This question is more towards clinical development.
Sir, actually, AMP are present in our body also. So I think we can boost our immunity system.
Okay. No, my question is more towards like in terms of clinical development, like how can we reduce the cost? Is there anything as a mechanism like when we, as an microbiologist, actually, we have to even think about modification and even making it more bioavailable actually for the part, because this is what a concern which is growing globally. I like your topic, but in terms of the viability of this topic to get accepted is something which is difficult to understand. That's what was my question. Yeah. Thank you.
No, sir, I don't know any other way that can reduce the cost of AMP.
Yeah, you can take it at your note. No need if you're not. You can take a note
Sir,
of that.
even
No need.
now.
Yeah, you can
Yes,
take
sir,
a
yes, sir,
note of
yes, sir.
this question. Nothing ahead, because this is something like when we are working towards clinical development, we need to think about the modification of even producing the HDPs like this. Okay, because we as a scientist make more a voice actually into the scientific community. Actually, we need to even think about the larger scalability of this production of this HDPs. Okay, yeah.
Okay, I will note it.
Yeah. Dr. Bharti?
No question, ma'am.
Thank you, ma'am. Thank you, sir. I now call upon Mohammad Anam Shafiq Ansari. Judges, she is a PhD category student, presenter.
Hello. Am I audible?
Yeah, I'm so sorry, sir. Let me
No
correct
issues.
myself. Yeah. Continue, sir.
Okay. Just a minute. I'm sharing on the screen. Just a
Yes,
minute.
sir.
This is not- Okay. Yeah, here it is. Share screen.
Also switch on your camera, please.
Yeah, ma'am. Just
You're loading the whiteboard.
Ma'am, just a minute. Actually, this is my first time to use the
Yeah, no problems.
Because previously I worked within Google Meet.
Yeah, no problems. Shut this, close this window. Yeah, go to the presentation.
Directly presentation.
You share screen. Do you see share screen at the bottom where a top arrow, upwards
There
arrow
is an
is there?
option. There is an option share screen.
Yeah.
But when I'm clicking on, okay, here they open up these settings. Okay.
Yes.
Okay. Start now.
Yeah.
Now the screen is visible.
Yeah, but you have to go to your presentation now.
Yeah.
Keep on moving. Yeah. Yeah, one more. Yeah.
Is it now visible?
Yes.
Okay. So first
You
of all,
can
good
auto
afternoon.
rotate. Can you auto rotate your phone?
Okay. Just a minute. Auto rotate.
Is it not there?
Click on portrait, sir. Click on portrait.
Yeah. Just a minute. Portrait. Yeah.
Yeah.
Thank you so
Now
much.
fine. Just tilt your phone now.
Okay. Now. Okay.
Yes.
Now.
Go ahead.
Okay. Thank you so
You
much
have
for
to switch on your
guiding me.
camera. I think your camera is off.
Camera.
I mean,
Ma'am,
mobile, it's not
when
possible.
I'm staring on the screen, I think so. That's why the camera has been shut down.
Okay. Please go ahead.
Is now visible camera?
Camera is visible. Yeah.
Okay. So first of all, good afternoon, everyone. As I am Muhammad Anam Shafiq Ahmad Ansari, ठीक है, from the Department of Microbiology, R.K.T. College, and today my topic is on the in silico perspectives on the probiotic antimicrobial peptides against the pathogens target. ठीक है. So, as we all know, in an introduction part, the antibiotic resistance is one of the biggest global health challenges today. As the misuse and overuse, there is an overuse of an antibiotics which leads to the several pathogens to be, have an emergence. And to address these issues, we have an probiotic derived antimicrobial peptides or the AMPs, which can be a promising alternatives surely. So there is an in silico approaches help to predict the peptide protein interaction. So there is an efficient method. Before we proceed with the wet lab validation, we have to move towards the dry lab. So such as called in today's generation, we call as a bioinformatics. Next, what is the need to do this study? Antimicrobial resistance threatens the effectiveness of existing antibiotic. And secondly, the probiotic AMP are naturally occurring, selective, and it is a safe antimicrobial agent, making them the suitable candidates. So finally, computational docking offers a rapid method to predict interaction between the AMPs and between the bacterial proteins, different kind of bacterial proteins. So by using up the computational tools, we can identify the promising therapeutic candidates before investing into the wet lab validations. Now the question arises, the gap analysis, and despite this potential, there are some clear gaps as well. Most computational studies still now focus on the small molecules only, okay? Not a peptide-based therapeutics. But very few studies have explored bacteriocins such as niacin A and pyridoxine against the clinically important bacterial targets. So this shows the any kind of a lack of data which slows down the transitional of these peptides into the clinical applications. But only limited work integrates peptide modeling tools like I use the HepepDoc with the docking against the multiple pathogen proteins. Now the time comes of an aims and objectives. The primary aim of this study was a threefold: to model the probiotic antimicrobial peptides such as niacin A, pyridosine A, PA1, and plantaricin E and enterocin E. So to perform the molecular docking against this such three pathogen proteins, so there is an sortes A from the Staphylococcus aureus, and there is an one T2P from the PDB data bank. So I uploaded here I think so this is an PDB data bank link or we can say a short code for the sortes A from a Staphylococcus aureus. And then we move towards the DNA gyrase B from the E. coli and lesser R from the Pseudomonas aeruginosa, alias R, which is an 31x3. So to analyze the docking interaction to assess this therapeutic potential of these AMPs, we have a target protein. So these are the target protein. This is an image, actual image from the PDB data bank. So there is an sortes A and a DNA gyrase, and then we have an LSR. Next, so sortes A is a virulence factor found in the Staphylococcus aureus. So it helps the bacteria attach to the host tissues by anchoring the surface proteins to the bacterial cell wall. And by inhibiting the sortes A, we can potentially reduce the ability of the bacteria to colonize and surely and to minimize the infection as well. And we can inhibit as well. So it is a key protein target. Then we comes towards the DNA gyrase. So it is an essential enzyme in the E. coli, which helps to replicate the DNA and transcription as well. It is regarding the central dogma, transcription, translation. So it is a well-known target for many antibiotics, such as fluoroquinone. Next, antimicrobial peptide can successfully bind and inhibit the DNA gyrase B as well. Next, LASR is a transcriptional regulator. Now, what do you mean by regulator? Which switch on and switch off the transcription and translation. Yeah, and regulator in the Pseudomonas aeruginosa. So, which is the bacteria communicate and regulate the virulence factor production. So, by targeting up the LSR, so antimicrobial peptides could potentially disrupt communication in bacterial communities and reduce the pathogenicity. Yeah. So in the methodology, the peptide sequence were retrieved from the NCBI database, and the protein structure were downloaded from the PDB, a protein data bank. When it comes to the docking tool, for the docking tool, we use the HepepDoc, and advanced version is, I think so, there is an HDoc server which predicts the peptide protein interaction. And for the visualization, which can be done by a PyMol, by analysis done by removal of an water, hydrogen bonds, and hydrophobic interactions as well. Now, in a
Mohammed,
final
you have to
result,
start the conclusion, huh?
direct conclusion.
No, yeah, I mean, you have some few seconds more. If
Okay,
this is
okay,
the
okay.
slide, then you can continue.
Okay. In a result part, I can conclude that such the probiotic AMPs such as lysine A, pyridosine, plantaricin, and enterocin demonstrated the promising docking interactions with the bacterial target. As we see the binding energy. So in the case of an binding energy, if lysine A has been targeted on a DNA gyrase B, so it is in minus 8.6, which shows the strong binding affinity. When it comes to the enterocin A, which is in the last, it shows the minus 7.5, which is a slightly weaker, but it still shows a good promising result. But in the case of an sortes A, it is in 7, sortes A, it is in the 7.9, slightly good from a DNA gyrase B, and in the case of an 8.1, it shows the drug-like molecules because plantaricin A peptide against the target protein. Then actually this was an image from the HepepDoc. So peptide lysine A on a target protein DNA gyrase B and pyridosine PA on the sortes A, and there is an plantaricin A on the LASR and DNA gyrase B enterocin. So
Kindly
in a
wind up.
conclusion, okay. This is in reference.
Dr. Bharati?
Yes. You write PEP fold modeling, right? So how do you decide which PEP fold 3 model to use for the docking? Can you hear me?
Sorry, ma'am.
How do you
Yeah, can you please repeat the questions? How do you
decide which PEP fold 3 model to use for the docking?
PP43 model used for the docking?
Yes.
Okay, so basically, I use the HEPEP doc server.
Okay. But I think slide number 3 or the 4 you wrote the PEP fold 3 model.
Slide number 3.
Check this once again.
Just a minute. 3. Because I tried the PP Fold 3, but because I can't be able to continue it, because
Yeah.
PP Fold 3 I can't be able to handle it. So after discussion, I use the HEPEP doc.
Okay, got it. Thank you.
Dr. Mankar?
Yeah, Mohammed.
Yeah, sir.
Yeah. Your discussion is more towards this AMPs, which are around like engineered probiotics, right? And
Yeah.
you are more towards focusing on this therapeutics part, actually, which is trying to state with lot of the efforts that you are trying to project. But what I find is, as my first impression, as you are a doctoral scholar, there is this research rigor which is lacking. So my first question to you is like, uh, in what way actually you find this translation into the clinical part, where you're as a research actually taking this potential responses of AMPs towards probiotic, and how would you create the clinical applications of it?
Actually, sir, as I'm doing the PhD in a current scenario, so my PhD is based on the probiotic only. And in the probiotic part, as we all know, the probiotic is then very good for our intestine and many more such things. So in my proposal, I've had the bioinformatics work because there is then various pathogens in our intestine, due to which we can't be able to absorb the various nutrition from our food source. So I'm trying to do key by using up the bioinformatics and by using up the probiotic, where I can stop the dangerous organisms in our intestine, where I stop and even I kill the those dangerous organisms, because sometimes we eat foods from our local food corners and हमें पता नहीं होता है कि कितने organisms deadly they came into
Okay.
our intestine.
Yeah, okay, I got it actually, because this is something like, now you cannot convince with word, you need to show with results. Okay, my next question is like, what are these AMPs actually that you are stating, which are producing probiotics? Are they able to transfer the antibiotic resistant genes? And if though they are able to, what are the strategies for mitigating the risk?
Sir, actually, we have done just only a dry lab experiment. We can't be able to say कि it is 100% sure कि that the organisms, or we can say कि the proteins, it will be antimicrobial resistance.
Yeah, but
So by doing of this,
yeah, please proceed.
so by doing up the only the wet lab experiments such as Kirby-Boyer method and various such kind of methods, we can predict
But this can be
organisms
done
and
even at master's level. Why we have to do even a scholarly study for going because this proposition should be prepared prior hand actually. Okay, fine, no head, no good actually. Okay, yeah.
Thank you, sir. Thank you, Mohammed.
Welcome.
I call upon Johanna now. Johanna Gonsalves. Yeah,
Ma'am, am I audible?
yeah, you are.
Ma'am, is my screen visible?
Yes, it is put into presentation mode, slideshow.
Is the slide changing?
Yes, please start.
Thank you, ma'am. Yeah. Good afternoon, everyone. I'm Ms. Joanna Gonsalves, a research scholar of Cluster Research Center, Government College, Khandola from Goa. I'm working on the topic exploring plantarisins as PBP2A inhibitors in MRSA through docking studies. So, 1 minute. Yeah. So, as we already know that S. aureus infections are a major cause of hospital and community acquired infections. They also not just cause skin infections, but they can also cause life-threatening conditions. In 2019 also, there was one report wherein it was stated that 9,40,000 deaths were caused by S. aureus, which were more than HIV/AIDS. And the main strain in this S. aureus, which causes the deaths, is the methicillin resistant S. aureus because it is resistant to most of the antibiotics. Yeah. So as you can see here in this figure, normally the bacterial cells have this penicillin binding protein 2, to which the penicillin antibiotic binds, and the peptidoglycan layer synthesis is stopped, and that's how the bacteria is killed. But in MRSA, they have this PBP2A modified protein wherein there is no binding site for the antibiotic. So the antibiotic cannot bind properly and it becomes resistant to the antibiotic. Also, the Lancet has said that in 2021, deaths due to AMR were 1.14 million. And now, in 2050, it can go up to 1.91 million. So, we require novel inhibitors for this protein. I'm working on fermented foods from Goa, and I have isolated Lactobacillus plantarum and found it to be having some antagonistic activity against many pathogens. So that's why I decided to check for plantarisins whether they can have some effect on this PBP2A protein. So as we know, plantarisins, they are antimicrobial peptides which have wide range of antimicrobial activity. The main activity that we already know is the permeabilization of the cell membrane. So I wanted to know whether it can also be having some binding to PBB2A and cause some changes. So a similar study has been done by Sinolia, but with antimicrobial peptides, which they have synthesized. So there is no study yet with plantarisins. So keeping this in mind, these were the objectives that I selected. Firstly, to screen the plantarisine peptides against the PBP2A protein using docking. Then secondly, to analyze the binding interfaces and interactions. And thirdly, to assess the binding affinity of penicillin to docked clusters. So if I am getting some binding, whether it could cause some changes with how penicillin binds to that protein. So my methodology was as follows. Firstly, I got a target protein PBB2A from RCSB PDB database. You can see the image here. The ID was 4CJN. I also got the plantarisine peptides from Uniprot. I got the FASTA sequence, also got the protein from PDB, and which I did not get, I took it from AlphaFold. So these were taken and then they were prepared using Pymol, that is removal of the solvents and so on, and energy minimization with Pyrex. Then I did the docking with Class Pro, and then I chose the top 5 leads, which gave me good binding activity, and I refined them with header. Further, I did 3D visualization and 2D interaction. And also I wanted to check whether this cluster has having some changes when penicillin binds to it. So I did docking along with penicillin G in Autovina. So these are the docking scores that I got from Class Pro. As you can see here, it is shown in purple. The good ones are shown in purple. The highest docking I got with KL, then NC8 and so on. Then I did head dock redocking, and for the 5 top leads, so you can see KL showed the highest head dock scores. Then you could also see the different interactions, van der Waals interaction and so on. And the clusters performed and the best clusters having this Z score were selected. So from here, these 5 were the best plantarisans. These are the clusters that I have got: KL cluster, J cluster, S, KN, NC8 clusters. These are all the plantarisan and the PBP2A clusters. So late after that, I did some interaction analysis using LigPlot plus to check what are the interactions that are there. And I saw mainly this hydrogen bonding, which is there between few of the amino acids. I'm not mentioning all, but it is there in the figure. And also we have hydrophobic contacts between them. Plantacerin KL showed one major groove, whereas the other plantarisans like J, S and NC8 showed a spread out binding. Then for my further inquisitiveness, I wanted to know whether it is changing the binding with penicillin G. So I did docking with penicillin G and the PBP2A protein itself, and I got the binding affinity of -6.1. Then I also did the docking of penicillin G with the other clusters, and I saw there was a slight increase from -6.1 to around -7.4, which was the highest for the KL plantarisane, which was till now showing good results. Then I also did 2D structure analysis. Here I saw that the penicillin PB2A showed a more spread out interaction, wherein there were more of hydrophobic bonds, but the one with penicillin and KL showed more hydrogen bonds. So this has to be still further studied. So to conclude everything, I can say that the plantarisane peptides that I checked for binding gave me good binding potential. Secondly, the plantarisane KL showed me the best docking. Also, penicillin binding had improved to this plantarisane protein clusters. And maybe the probiotic derived metabolites like this plantarisans, they can enhance the efficacy of conventional antibiotics by interacting with such resistance associated targets like this PBP2A. So this was my conclusion. So maybe this plantarisans can help have some synergistic potential, and it has to be further explored. These are my references. Yes, thank you.
Dr. Mankar. Mankar sir, are you there? Dr. Bharati, please go ahead and ask question.
Yes. Hi.
Hi,
Can
ma'am.
you explain the significance of targeting PB P2A in MRSA and why plantarisine KL1Y was chosen for the molecular docking study?
Oh, ma'am, I forgot an important part. Actually, I have screened many plantarisans because I was interested in Lactiplanti, Bacillus plantarium. Those are the majority strains that I have isolated. So I tried around 21 plantarisans, and from that, this 5 were the top ones, and KL showed me the best binding activity. So I went further with that.
So firstly, my suggestion is you start explaining PBP2A role in MRSA resistant to the beta-lactam active antibiotic, right? And
Yes,
then
ma'am.
discuss the plantarisine. Okay.
Yes, ma'am.
Okay. Thank you.
Yeah. Thank you, ma'am.
Thank you, Joanna. Thank you, ma'am. Now I call upon Geeta Ojas Purohit, madam. Judges, she is from faculty category. Ma'am, kindly present.
Yes. Am I visible?
You are visible, not your screen yet.
Yeah, 1 second. Now?
Not yet, ma'am. Yeah, it's starting now.
Yeah, 1 second.
Yeah,
Is it in presentation mode?
not yet.
Okay. Now is it?
Yeah, ma'am.
Yeah.
Kindly go ahead.
Yeah, thank you. So good afternoon, everyone. I'm Geeta Ojaspurai from SP College, Pune. So the title for my research is Structure Prediction and Docking-Based Screening of Anti-Cancer Targeting Conserved Domain, especially in oncoproteins. So moving forward to the aim of this project, to design and evaluate the potential activity of curcumin targeting conserved domain of key skin cancer associated oncoproteins. For that, we select the conserved domain. So in that, EGFR, BRAF, PD1 or PDL1 and STAT3 are the conserved domain for the skin, particularly cancer. So for a melanoma, we select the PDL1 as there is no such work, dry lab work in the PDL1 sector using a structure-based computational drug design discovery approach. So for objective structure-based molecular docking, dynamic stimulation and ADMT studies, so we, ADMET studies specifically performed to evaluate the drug candidates absorption, distribution, metabolism, excretion and toxicity, which are critical factor for its safety, efficiency, regulatory approval in drug development. So ADMT studies of curcumin with the conserved domain for skin cancer associated oncoproteins to access the binding affinities and interaction profiles. We studies the SSDD powerful approach for the 3D structure of target 3D structure of target protein to identify optimized small molecules with the high efficiency and which show the efficiency against the disease-associated targets. So curcumin is a natural bioactive compound, exhibit the promising anti-cancer effects by modulating multiple signaling pathways. Okay, so this study aims to evaluate curcumin's potential to target the conserved domains of key skin cancer-associated oncoproteins. So for that, we include the PDL or PDL1 using the structure-based computational methods. For that, we do some I do some literature reviews, so that I found the gap analysis. So most studies on curcumin inhibiting PDL1 in cancer have up to focused into the in vitro, in vivo or wet lab experiments. So there is no such work in a dry lab. So for that, we identifying the conserved domain and perform the virtual screening, ADMET analysis, followed by the future. In future, we are going for the MD stimulations methodology. For that, specifically, first we search for the target melanoma. Here you can see that. So then first we perform the literature review for the natural drug to find out the natural inhibitors of the PDL1, which is 5045. After doing the literature review, I find that there are the research gap for there is very there is a very less dry lab study has been done on the curcumin and PDL1. So I downloaded all the structures of curcumin from the PubMed. After that, I convert them into the separate SDF format, and with the help of OpenBabel, convert them into the PDB QT format. After we performing the ADMET study to assure the safety, efficiency, and regulatory approval in the drug development, I use the ADMET Lab 3.0 for ADMET I would say ADMET research. For ADMET, I have used the Python script for filtering the compounds. After that, I perform the after performing the ADMET, I get the direct ligand which passes the parameters of the ADMET. After that, I selected the PDB structure, that is 5045, which have take second for the multi-ligand docking, and after performing the multi-ligand docking, I get the 6 compound which shows the higher binding affinity or energy with a 5045. So till now I covered this part, and I found this 6 ligands with the high affinity energy or affinity with binding energy with a 5045. Then with the help of PyMol, I convert those into the 3D format, ligand protein interaction. And with the help of BioVR Discovery Studio, I retrieved the 2D interaction with ligand and proteins. So here
Madam, kindly go to conclusion.
Yes, here I conclude these results suppose to the therapeutic potential of natural compounds, specifically curcumin, in the treatment of skin cancer, which is melanoma, a very aggressive subtypes for which there are few available treatments because of ligand attractive ADMET profile, low intermolecular energies, and consistent cross-platform docking scores. Above all 6 excellent candidates ligands for the additional in vitro and in vivo validation. Thank you so much.
Dr. Mankar. Dr. Bharti, please go ahead.
Yes. Can you tell me the curcumin binding model and its functional impact on the PDL1 interaction?
Ma'am, I didn't get your question. Can you please repeat?
Okay, fine. What are the specific molecular interaction between the curcumin and the PDL1?
Curcumin and PDL1.
Yes.
So ma'am, there are some, so as you can see here, there are the there are 2 proteins. First is a PD and second is a PDL1. So we select the PDL1 which already have the receptor sites. So those ligands are directly bind to that receptor sites of the PDL1 and we get these structures and the affinity, higher energy level.
Okay. So from this diagram, can you tell me the curcumin directly form bonding with the PDL1? So which amino acid residues available there?
Sorry ma'am, I have to study that part.
Okay. Please study. It's important part. Okay.
Yeah. Thank you so much, ma'am. Thank you, Geeta, ma'am.
Thank you, ma'am.
Now I call upon Ms. Palak Chaudhary. Judges, Palak Chaudhary is from PG category. Geeta ma'am, stop sharing, please.
Good afternoon, everyone.
Palak ma'am, just a minute. Huh? Yeah, start please. Switch on your camera and then share. Yeah.
Ma'am, is it fine?
Yes. You can start sharing now. Your screen is visible now. Put into slideshow. Yes, start please. Yes, start please.
Good afternoon, everyone. So the topic for my presentation is in silico and computational advances in Alzheimer's disease. So basically, it is a comparative study for dual target approaches and novel therapies. Before going further, let me discuss about the Alzheimer's disease. What is it? Like in the picture, we can clearly differentiate between the normal side of the brain and the Alzheimer's brain. The ventricles are enlarged. There is a atrophy in cerebral cortex, and it is a neurodegenerative disease that affects the brain function, and the 2 main culprits of the brain of the disease is that tau protein and amyloid beta protein. So it leads in memory loss, dementia, and in cognitive decline. So the current treatments that we have are very limited, and they only treat the symptoms. So there is an urgent need that this disease should be cured. So as I mentioned, there are two main culprits, but the other factors that are also responsible in Alzheimer's disease are oxidative stress, metal ion dysregulation, mitochondrial dysfunction, neuroinflammation. So there are uh, multiple mechanisms through which Alzheimer's can be caused, but there are very limited targets that we target, or there are only single target interventions. That's why there is a limited clinical efficacy. So let me discuss how the Alzheimer's is caused. There are two proteins, as I discussed, amyloid beta. So it is the small peptides. When amyloid precursor protein gets cleaved by beta secretase enzyme or gamma secretase, we get amyloid beta, and the accumulation of amyloid beta is and yes, accumulation of amyloid beta is caused, that is outside the neurons, and the tau proteins is the hyperphosphorylation and aggregation. So tau protein is the scaffold of neurons, so that gets disintegrated between them and forms a sticky like structure inside the neurons itself. So these are the two main pathways that we can say that Alzheimer's disease is caused through. Now, these are the structures that I took from PDB, cryo-EM structures of tau filaments of Alzheimer's disease, and 3D structure of Alzheimer's, amyloid beta 142 fibrils. So, ma'am, like I'm showing the salt fibrils structures of two key proteins of Alzheimer's pathology. On top is the tau fibrils, and the below is amyloid beta 1. And these are some of the molecules that are responsible for treating both the disease. So we can see that the quercetin, curcumin, and berberine are the three molecules that shows the strong inhibition for BACE1, and as well as shows the inhibitions for tau fibrillations, whereas EGCG and resveratrol shows moderate inhibition, but also has a good sorry ma'am, and that has a good suppressions for the tau fibrils formation. Ma'am, this is the VHH Z70 interactions of BHF7 tau peptide. So VHS Z7 is a basically nanobody that is interacted with the tau protein. We can see that the nanobody is bound with the protein structure, and the PHF6 is a hexapeptide that forms beta sheet spine of tau fibrils, making it a good it makes a good docking surrogate. So by targeting PHF6, we can aim to interfere the tau aggregation. And BACE1 binding, it is the binding of BACE1 with the ligand called quercetin. I performed this docking and I got a good docking score of 8.2.
You'll have to now go to conclusion fast.
Yes, ma'am. To conclude that both the proteins are very important for treating Alzheimer's disease, and the common factors that between both of them are like they both promote hyperphosphorylation and aggregation, and there are many other common factors. They show single target drugs and dual inhibition address downstream tau aggregation. So I can say that by targeting both the pathways we can treat Alzheimer's disease. And yes, ma'am. Thank you.
Dr. Mankal? Dr. Bharti, please go ahead.
Can you tell me the name of software for the silico analysis?
Ma'am, for docking, Autodock Vina and Pyrex can be used. Pyrex is used for like batch docking, so like we can do a multiple screening through it.
Okay, give me the workflow for the Alzheimer disease
Ma'am,
related to molecular docking only.
yes, ma'am. Ma'am, protein should be extracted from PDB database, and then it should be the protein extracted. We should remove the water molecules and add hydrogen molecules, polar hydrogen. And after that, the ligands from the PubChem database should be extracted. And for docking, we do use Autodock tools to see how well that structure is binding by looking at the energy binding affinities.
Okay, thank you.
Nishad, sir?
Okay, ma'am. No questions from my side.
Thank you, sir. Thank you, Palak. Can stop presenting. Now I
Thank
call
you.
upon Naushin Chaudhary. Naushin. Patil sir, kindly
No, ma'am.
confirmation.
No, ma'am, she is not.
Is not there.
Waiting.
We'll go on to the next. Mr. Hassan Raja.
Hello, actually, ma'am, Naushima and Hassan messaged me that they are in the college. Their exam is going on.
Okay, okay. So I'll go on to Zoshi Pooja.
Ma'am?
Pooja Zoshi. She's a faculty.
Yes, ma'am.
Yeah, ma'am. Switch on your camera, ma'am, and start presenting, please.
Can you see me, ma'am?
Screen is about to start, not started yet. Yeah, I can see the screen, but it's somewhere else, not the presentation.
Yes, ma'am. I will open.
Your presentation can be seen now. Put into slideshow. Yes, ma'am. You can continue.
Myself, Pooja Joshi. I have a PhD student at Balbim Arts Science College, BA. My project title is pyrrolipidamide derivative docking study targeting PI3K and AKT proteins in MCF7 human breast cancer cell line. Introduction. In many Western nations, breast cancer remains leading causes of death among the treatment as well as prevention on much of laboratory work and clinical over the 30 years. Pyrrolipid shows various biological properties, including anti-cancerous, antibacterial, antifungal, anti-inflammatory, antiviral, and anti-diabetic properties. Among these, if pyrrolipid is showing anti-cancerous activity, that's why I'm selecting pyrrolipyrimidine for my project. In this, I'm targeting PI3K and AKT proteins of the MCF7 MCO7, this study investigate the binding affinity and interaction of protein selector pyrimidine cell derivatives with PI3K AKT proteins, both of which are crucial target in a breast cancer therapy. Aims to identify suitable drug from pyrrolipidemine derivatives against MCF7 cell line to add a new drug in a breast cancer therapy. These are ligands which I have used for the study. These ligands I am take from the ChemDraw software. These are the two proteins which I have targeted, PDBID 3HHM and 4EKL. These I am taking from RCSB database. Research methodology: protein downloaded from RCSB database, protein cleaned by removing water molecule and ligand by using PyMol software, ligand smiles code get from PDB Chem and ChemDraw software, which are not available in the
Your voice is breaking, ma'am. Yeah, ma'am, your voice is breaking. Can't hear you. Pooja, ma'am. Judges, it seems she has internet problem. We'll give her a minute. Pooja, ma'am, if you can hear us, rejoin. In the meantime, I'll call. Yeah.
Yeah.
I'll call the next, na?
Yes, yes.
Yeah. In the meantime, I'll call the next participant, Aishwarya Saha. Ma'am, please unmute yourself and switch on your video.
Ma'am.
Yes.
Am I
Slide
visible?
is seen. Yeah, you're visible. Just put into slideshow.
Yeah, okay. Okay, so
Begin.
good afternoon. I am Aishwarya Shah, Assistant Professor from Rainbow University. Today I'm going to present on the topic Insight into the Potential Therapeutic Efficacy of Nano Hybrid in Biomedical Application and its Molecular Docking. So as we know that breast cancer remains as one of the most prevalent and lethal cancers worldwide, with its triple negative type being particularly aggressive and resistant to conventional therapies. So through this research using nanotechnology, this offers a promising solution by enabling targeted delivery and multimodal therapeutic actions. So in this study, I have taken gold nanoparticle and sodium oxide nanoparticle and exploited both of their anti-cancer properties and merged them together to form a nano hybrid and assessed its therapeutic efficiency against TNBC cells, triple negative breast cancer cells, and also studied its pathway analysis and molecular docking specific proteins. Well, we have synthesized this nano hybrid using green synthesis, which is a very eco-friendly method and also environmentally sustainable. Then we have, for results and discussion, we have studied physicochemical characteristics via UV visible spectroscopy, FTIR, and XRD, which all of this has confirmed the crystalline structure and the formation of nanohybrid. For in vitro cytotoxicity study, we have done the cell viability assay using by a dose-dependent manner from 10 microgram to 200 microgram per ml, and for higher dose, we have seen percentage of cell viability decreases. And for in vitro cellular uptake, we have taken the RITC labeled nanohybrid gold Syria nanohybrid, and it is clearly seen that there is significantly higher fluorescence in the cell cytoplasm region, which obviously indicates better cellular uptake. For the membrane potential study, we have done the JC1 imaging, and here also it is shown that there is a significant drop in fluorescence intensity from red to green, which signifies loss of membrane potential in the triple negative breast cancer cell on nanohybrid treatment. And the anti-cancer effect, anti-migratory effect of nanoparticle on cancer cells has also been studied. Here also it is shown that the after treatment with nanohybrid, there is significant decrease in the number of lamellipodia and filopodia, which are the main cytoskeletal elements that helps in the migration of cancer cells. In the apoptotic study, we have seen that there is significant increase in the number of apoptotic cells on treatment with our nanohybrid. For Western blot and pathway analysis, we have seen there is upregulation and downregulation of three main cancer cell signaling proteins, which are NF-kappa B, which is downregulated, and PAKT, which is downregulated again. But P10, which is a tumor suppressor protein, is being upregulated after nanohybrid treatment. This demonstrates nanoparticle mediated suppression of oncogenic pathway and induction of apoptosis. For molecular docking, three proteins which are again very important for the cell signaling of cancer cells, P53, NF-kappa beta, and kinase protein have been taken. P53 being a tumor suppressor protein, NF-kappa beta being a cell survival and proliferation protein, and kinase being another regulatory signal transduction protein. So before molecular docking, we have done the Ramachandran plot analysis. And here it is shown that all of the three proteins that we have selected demonstrate good stereochemical quality, and a higher percentage of the residues are in the favored region, which confirms structural reliability for docking simulations. Then for the molecular docking study, we have used the software Autodoc Bina, and we have evaluated the binding interactions of our nanocargo, which is the gold Cydia nanocargo, and 5-fluorouracil, which is a well-known anti-cancer drug. And we have checked that against the proteins P53, NF-kappa beta, and kinase protein. From the interpretation, we can clearly see that if 5-fluorouracil, which is an anti-cancer drug, shows stronger binding, and our sodium oxide nanoparticle also shows consistent interactions. This confirms specific interactions between our nanocargo and 5-fluorouracil with regulatory protein involved in the triple negative breast cancer progression. And this result suggests a synergistic mechanism for further nanocargo that we can deliver for targeted delivery. We can incorporate certain drugs inside those our nanocargo and enhance the targeting ability of the drug. So to summarize my entire study, we have developed a green sustainable methodology for synthesis of a nanohybrid, and then we have studied the several cellular uptake and apoptotic study. After that, we have also done the in silico molecular docking study for against the TNBC. So this all of these findings provide a solid foundation for further preclinical evaluation and development towards personalized and precise treatment strategies for triple negative breast cancer cells. And lastly, to acknowledge, this is a collaborative work which I have done with all my colleagues. So thank you for all your patience.
Dr. Bharti?
Yes. Can you compare the molecular docking of gold nanoparticle with the organic nanoparticle?
Yes, ma'am. So actually, we have done this with rare earth metals because by comparing, we have found that the rare earth metals inspire. It also serves as nanocargo, but also it also has anti-cancer properties like redox properties, ROS modulation. So that is why comparatively, these are the nanoparticles that are showing better results than the organic.
As a suggestion, can you try, please, in the future, try the PGLA, that is the polylactic glycolic acid. This is also good one. Okay.
Okay, thank you.
Thank you.
Thank you, ma'am. Thank you, Aishwarya, ma'am. Zoshi, ma'am has joined. We can tell her to continue her presentation.
Yes, ma'am.
Yeah. Yes, Zoshi, ma'am, please continue.
Can you see my?
Yes, yes, yes.
I will continue when I'm.
Go to the slide where you stopped.
Yes. These are the ligand which I have selected for the docking. These are the two proteins which I have selected for the docking, PI3K and AKT proteins of MCF7. Protein downloaded from RCSB database, protein cleaned by removing water molecule and ligand by using PyMol. Ligand Smiles code get from PubChem and ChemDraw software. Ligand Smiles code converted to SDF MDL mole format using OpenBabel web tool. These ligand converted to PDBQT format using Pyrex software. Pyrex software utilized for molecular docking. Result downloaded in CSV file, later converted to Excel format for data analysis. These I get the result for the PI3K protein. These are the different ligands which I have docked with the PI3K. I get the most result at minus 9.6 binding affinity. Results for AKT protein, I get minus 8.2 is the highest binding affinity, lowest binding affinity. These are the 3D structure of ligand 5 with 3 HHM and ligand 5 with 4 EKL. Ligand 5 gives the best result, best binding affinity. These are the 2D interaction of for PI3K with 3 HHM, ligand 2 and ligand 5. Results for 2D interaction of AKT, 4EKL, PDB ID, ligand 4 and ligand 5, for ligand 4 and ligand 5. Conclusion, all the ligand docked with PI3K, 3HHM and AKT4 EKL. Among all the ligands, ligand 2 and 5 shows good binding affinity with PI3K, 3HHM protein, minus 9.1 and minus 9.6 respectively. Ligand 4, ligand 1 and ligand 5 shows good binding affinity with AKT protein, 4 EKL protein, minus 8.2, minus 7.1 and minus 7.2. In this, ligand 5 gives good binding affinity with PI3K also and AKT also. Future study some novel ligand to be docked with this protein in search of new drug molecule. These are the references where I'm taking the 3HHM protein, 4EKL selected, and for the MCF pyrrolipidimine derivative from these two papers. Thanks to R.K.Talreja College to providing me this opportunity. Thank you.
You're welcome, ma'am. Dr. Bharti.
Which software used for the docking for pyrrole pyrimidine?
Pyrex software user.
Why didn't you use the Schrodinger software?
Actually, ma'am, I am from organic
It was
chemistry
a
and
very
just
nice result.
yes, ma'am, I am from organic chemistry background. Just I learned these tools, and I will learn more for this and get a very good result in next.
Okay, okay, ठीक है. Which interaction types are mostly important for the pyrrole pyrimidine and their target binding?
Sorry, ma'am.
If you've done any interaction
Yes.
with the of a pyrrole pyrimidine with their target, can you tell me the most important types of interaction within them?
Actually, pyrrolopyrimidine with the chlorine group, it gives good affinity with that proteins.
But that have the some residue, some stacking, some contacts, anything. Can you tell me?
It's a hydrogen bonding type.
Okay. Okay. Yes, thank you.
Thank you, ma'am. Thank you, Pooja, ma'am. I now call
Yes.
upon Sri Day.
Ma'am, can you repeat the name?
Sri Parna Day. SRIPARNA. She's there, sir.
Yes, ma'am. Done.
Yes, ma'am. Screen is visible, ma'am.
Yes, yes. Yes, ma'am. Please go.
Yeah. So, a very good afternoon. So, myself, Dr. Shriparna Dey, I'm from Brainware University, assistant professor. So, today my study is to emphasize the potential efficacy of that hydrogel material. So, that will be implemented in future for the AI integrated self-healing antibacterial strain sensitive application as an wearable healthcare devices for healthcare monitoring application. So, directly if I'm going to the hydrogel section, we all know that this is having a special property with some sort of stretchability, along with that some sort of biocompatibility. So you can make it, if we can make it in a responsive way, in such a way we can make it in stimuli sensitive way, so that we can future, if you can implement this hydrogel material as a strain sensing application. So here, moving towards the preliminary section, we have started with the synthesis of various type of nanoparticles. It is there in a various type of nano range for the functionalization. After functionalization, we are getting lots of antibacterial property. We can see it is an enhancement of wound healing efficacy, along with that enhancement of the stretchability and functionality in the biomedical application. We have studied the basic and basic study we have done. So especially we have focused here in the rare earth metal oxide, that is a cerium oxide. So we have studied the in vitro as well as the in vivo aspect of that. So these are the preliminary, we have done this preliminary study. Along with that, we have taken a different types of Caragen based material. So this material we have functionalized with a different types of quantum dots, and we have seen this is showing us some sort of optical transparency. Along with that, some sort of color change. We can measure it in a colorimetric way. This sort of film we have, when we have functionalized this with a stearic acid and quantum dot material, then we have studied this experiment. We have done this paper for the analysis of that prawn shell, that freshness monitoring application of the prawn cells. So this is the preliminary study we have done in the monitoring application. So these are already published one. Along with that, whenever we are focusing on various type of hydrogel materials, so different types of thermoresponsive property we are trying to generate there. So we have prepared a poly N-isopropyl acrylamide based PVA grafted polymers. So this are showing some sort of responsive nature. Along with that, when we are modifying this one with a lysine, so we have fabricated some sort of patch material that also showing some sort of color variation. So if we are focusing on that particular thing, we have grafted with a collagen, we have grafted with a lignin. Along with that, we have grafted with several types of antioxidant to enhance the antioxidant potential. Here, the major findings we have got in an in vitro study as well as in the wound healing application, we have studied the in vivo one. So we have done the ex vivo analysis of the mice model, and we have seen a significant result with diabetic control. If I'm seeing here, as compared to diabetic control, that treatment shows a higher mortality. Along with that, now my next phase of work is to validate that part. So I am trying to validate this kind of patches or this kind of hydrogen in the form of sensing application. So directly, if I'm thinking this sensing application, I will show the graph, I will show the video as well. So here we will see the changes is there if we directly focus on the integration part of AI. So Arduino we have used for that, and we have modulated in a breadboard. So directly, if I'm fabricating this thing in a smart bandage, so automatically with respect to pH and automatically with respect to moisture and temperature, it is changing constantly. Along with that, if I'm focusing on the various type of functionalization part, suppose in case of strain sensing application, we have done a hydrogel based system. So here also we have set up a real-time monitoring application. There we have done this to analysis. So with a wrist bending and wrist stretching, so we have seen this type of modulation. So hydrogel and with Arduino we have used. After that, serial communication we have connected with a Python, and real-time monitoring we have seen. So directly we can implement this part in a wearable sensing application or as a wearable device. So this is my expected deliverables. This materials, whatever we are preparing, these materials are highly promising hydrogels that will be offering a very good electronic skin devices along with that wearable sensing devices. And in future we are planning to do in a on-demand drug delivery that will be miniaturized as a smart bandage. So that will directly, I think, address this the diabetic wound healing or diabetic foot ulcer. This type of chronic wounds we can easily manage. Along with that, the precision medicine wise, no need to visit a doctor without altering the bandage, without altering the dressing for the recurrent infection. So we can do this type of material fabrication. In a future aspect, we can implement this type of material in a different types of health monitoring application purpose. So this is the future work plan. We are trying to focus on the dual responsive nature for the multifunctional way. Suppose pH, thermo responsive, light responsive as a whole. So this thing we are trying to develop. So I want to acknowledge this is a seed grant project. So already this is a few parts we've done here and few parts is there in University of Calcutta. So my mentor, Dr. Professor Dipankar Chattopadhyay, along with that, different other department, SSKM Hospital, IPGMR. Thank you.
Dr. Bharati.
Nice, really nice work. So can you tell me the how do you prepare the hydrogen, sorry, what do you use?
Hydrogen material.
Yes, hydrogen gel.
Yeah.
Which molecular docking applications or software use for
This
that
is,
purpose?
madam, this is not a molecular docking actually. This is
Yeah,
the part,
simulation.
yeah. Some
Simulation.
sort of simulation I have used
Yes,
for the
yes.
AI modulation. So this is the fully integrated way. So directly I am doing the transformation, the digital analog converter. So hydrogel, with that hydrogel material I have used Python. I have used a circuit. I have prepared the circuit, that one. So with the Arduino, I have shown this one. This is a model. This is a circuit model. So I am trying to develop key whether some sort of signaling, some if it is there in that is there, it's changing, its variation is there. So directly we can change it. Suppose one patient is in paralysis state. Suppose the patient is unable to move. For the Parkinson's patient, those who are not able to move, so if some sort of some signaling they were giving, some hand is movement,
Wait.
something they
You
are
use
trying to
the
tell. Yes,
Python and the AI simulation, right? Now, but
yes.
sometimes my suggestion is kindly use the Gromox and the Amber lamps. These types of the modeling polymer networks, which is giving the very nice stimulating practically releases. Okay.
Okay. Sure, ma'am. Actually, I am also myself, my background is a biochemistry. So anyhow, I have to club it with some sort of computational science or computer science section. So this is also a new aspect. Definitely, madam, I will keep in mind.
Yes. Thank you.
Thank you, Bharati ma'am. Thank you, Sriparna ma'am.
Yes.
Now I call upon Nirmala Pawar ma'am. Next participant. Nirmala Pawar.
Yes, ma'am.
Yeah, ma'am. Switch on your camera and start presentation, please.
Yes, ma'am. एक minute.
Till Nirmala ma'am does that, let me tell you, this is the last presentation of the day. If you all have not filled up the feedback links, please fill the form as soon as possible. Nirmala ma'am, can we assist you in any way?
Yes, ma'am.
Yes, ma'am.
Yeah, can we help in any way?
Ma'am, actually, I am presenting from mobile. So while sharing my screen, it is not possible to on the video, ma'am.
Yeah, okay. Please go ahead. Present.
Okay, ma'am.
Ranjana ma'am, did you wanted to say something?
No, ma'am, no.
Hello, ma'am.
Yes, ma'am.
While sharing the PPT slides, there is a problem. Can I share this with anyone in the present participants so that they can share the screen?
Yeah, that can be done. Please send it to whom you are sending, or you can send it to me. My number is there in the group.
Okay.
Please send on the WhatsApp. I'll do it immediately.
Okay.
AI group, my number is there.
Yes, ma'am.
Yeah, I have got it. I'll just download. I have presented it. Is it visible to you?
Yes, ma'am.
Yes, you can start, ma'am.
Okay, ma'am. Good afternoon, one and all. Good afternoon, one and all. I am Nirmala Amol Kumar Pawar, PhD student from R.K.Talreja College, Microbiology Department, Ulhasnagar, presenting this research paper, not a research paper, review paper with my friend, co-author Mr. Om Prakash Kadam, Digambar Bindu College, Microbiology Department, Bhokar, Nanded. We are presenting this review paper as Artificial Intelligence in Silico Drug Absorption, a comprehensive review. First, role of AI in first slide. Ma'am, next slide, please. First, role of AI in absorption prediction. Drug absorption plays a vital role in determining therapeutic effectiveness. However, Traditional conventional experimental methods are time-consuming, resource-intensive, and often contribute to a clinical trial failure. This is where artificial intelligence-based in silico model can analyze complex dataset, capture non-linear relationship, and provide accurate predictions to save both time and cost in drug development and absorption. Next slide, please. Next, fundamentals of drug absorption. Fundamental of drug absorption consist of physiochemical properties and biological barriers, in which physiochemical properties such as solubility, lipophilicity, and ionization, whereas biological barrier as intestinal epithelial and efflux transporters to evaluate absorption efficiency, parameter like CACO2 permeability and oral bioavailability. Next slide, please, ma'am. Role of AI in silico modeling absorption. Role of AI in silico modeling absorption such as machine learning ML, deep learning and hybrid approach. In machine learning, it supports vector machines. Machine learning plays a central role in AI-driven drug absorption, transforming new researcher to identify, design, and optimize new drug, and then follows ADMET, that is absorption, distribution, metabolism, excretion, and toxicity. Instead of wet lab absorption assay for thousands of molecules, machine learning rapidly screens chemical libraries for absorption-friendly candidates. Then deep learning. Deep learning, like convolutional neural network, predicts absorption directly from molecular structure. Then hybrid approach. Artificial intelligence with PBPK models, that is physiological based pharmacokinetics model for accurate simulations. Next slide, please. Database and resources for AI modeling. Database and resources. Reliable prediction requires database such as ADMET database, that is absorption, distribution, metabolize, excretion, and toxicity. ADMET database. Then CHEMBL database. Then PubChem database, which provides valuable information. Benchmark database are specially useful for validating CACO2 permeability and human intestinal absorption prediction. Next slide, please. Case study. Case study of artificial intelligence in absorption prediction. Recent studies confirm that deep learning outperforms traditional QSAR models. QSAR model is computational method that relates the chemical structure of molecule to their biological properties. This traditional QSR model or graph neural network shows excellent performance in prediction absorption endpoint. In pharma industry is also adopting AI enhanced PBPK platform like SimSip, population based PBPK model, then GastroPlus, which is mechanistic based absorption and pharmacokinetics model. This two advanced model pushing us closer to personalized drug absorption and prediction. Next slide, please. Challenges and limitation. Although there are advances, despite these advances, challenges persist, like data scarcity limitation, then variability in human physiology like age, gender, etc. Then deep learning models face a black box problem, make them less transparent for regular uses. Therefore, finally, integration of AI in real world clinical decision making is still limited. Ma'am, next slide, please. Future perspective.
You can see my screen is frozen, it seems. Just
Okay,
give me a
ma'am.
minute.
Next slide is future perspective in artificial intelligence for absorption prediction. In future perspective, there are self-supervised learning will leverage unlabeled data. Then digital twins may allow personalized absorption profiling. Then multiple task learning could predict absorption along with metabolism, distribution, and toxicity. Most important, explainable AI framework will ensure regular acceptance. Now, next slide, please. That is conclusion. To conclude on artificial intelligence in drug absorption modeling, AI has revolutionized in silico absorption modeling, improving efficiency, scalability, and accuracy, which challenges remain the integration of AI into pharmacokinetics, the potential to reshape drug discovery, absorption, and personalized medicine. With this, thank you to one and all. Next slide, please, ma'am. Thank you to one and all.
Yeah, it's okay, na? I stopped.
Okay, ma'am.
Yes, Bharti ma'am.
It's a review, right? Na? All modeling systems. Can you tell me how do molecular dynamic simulation contribute to understanding the drug absorption?
Sorry, ma'am.
How do molecular dynamics simulation contribute to understanding the drug absorption? You add all the simulations or all modulation theory, but not add the molecular dynamic simulation
Yes,
as
yes.
well as not add the Lipinski rule of the 5 in silico drug absorption studies.
Yes, ma'am. Dynamic simulation models moves, dissolves, and is absorbed over time in the gastrointestinal tract, helping predict absorption rate and bioavailability.
Okay, this type of the interaction between occurs. Okay. Interaction at an atomic level as well as the drug permeate the lipid bilayer which interact with the transporter. They are transporters like that. And for that molecular dynamic tools are Gromax and Amber. You didn't mention there.
Sorry, ma'am. I will learn for more damage for,
Yes,
for
got
you.
it, got it. Yes. Okay, thank you.
Yeah,
Thank you
thank you,
so much,
ma'am.
ma'am.
Participants and judges, my committee members, we will stop this particular session now. We will break for lunch and 3 p.m. sharp will join for valedictory and declaration of best paper awards. I request all of you to be on time at 3 p.m. so that we can finish in time. If you have not filled up the feedback forms, kindly ensure you have filled it up. Thank you. Ranjana ma'am, any other instruction?
Yes. No, I would like to thank Dr. Bharti.
Yes, ma'am.
And thank you so much for giving this valuable time to us and judging it so keenly along with Dr. Mankar. Even Nishad was there. He was handling many steps behind the camera most of the time. But special thank you to Dr. Bharti. And of course, Amina madam has coordinated this session so well. It was really an learning experience throughout this session. Bharti ma'am, you can express your views.
Yes, ma'am.
Okay, it's a nice and wonderful. Thank you for giving me the real opportunity as a judge. And really, it's an address as a judge as this remarkable conference on this silico structure analysis. And we have the witness as an inspiring arrays of the presentation, posters, and discussions on the different modeling. I really happy for that moment, and thank you, and thank you so much.
Thank you so much. Meena ma'am, you can conclude now this session so that
Yeah, ma'am.
we have to come for the valedictory function. If free, Bharti madam, please do join to see the result.
I have a schedule again. Thank you for that.
Thank you so much, ma'am. You all are asking, some of you all are asking for feedback form. Sir will post it now. Maybe in the group, sir. Kindly see to it that the form link will be posted in the group and you all please fill it up. Join back sharp at 3 p.m. so we can proceed with the valedictory. Thank you all.
Thank you.
Kuldip sir, can we share link here?
The link is already shared, I guess, in the chat box. I released it quite early today.
Okay.
It's sent at 11:56 and it is also shared with the participants in the WhatsApp too.
And they must have missed it then. Yeah, it is there.
Yeah.
In the chat box, the link is given. Kindly scroll.
Yeah, I'll reshare it again here also and as well as in the WhatsApp group also.
Sir has shared it again. Please check it out. It is there in the WhatsApp group too. Do fill it up. Patil sir, can we end the meeting now?
Okay, ma'am.
मॅडम आपण थोडा वेळ थांबणार आहे का?
आपण 3 ला जॉईन करूया ना, आताच थांबायचंय.
No, we'll take break now. Group will take break now, I guess.
Yeah.
Meena ma'am is occupied since I think 11.
So we'll join by 2:45.
Yeah. And meanwhile, we'll be active. Yeah. We'll be active on our WhatsApp group if anything else is required.
Okay, ma'am.
Chalo, then just end this meeting.
Yeah.